we are dealing from UPN. Got it. You can hear me clearly?
Boleh, Dr. Nalini, boleh dengar.
Boleh, okay. So my name is Nalini Armugam. I'm a lecturer from Sekolah Pusat Pengajian Sains Pertanian dan Bioteknologi and teaching here for 14 years. My field of expertise is agribusiness. Okay, allow me to continue with my topic. Today, my topic will be on management of food availability and supply chain management, which I'm going to focus on rice plant in Malaysia. Okay, rice plant is another name for paddy. Paddy. Okay, so it's a bit weird because I don't know my colleague in Indonesia can hear me or not. The audience from Indonesia can hear me or not. Definitely Malaysian student can hear me. I assume that. Dr. Salma, okay?
Yeah, okay. Loud and clear.
Loud and clear, okay. Okay, today my content will be, this is my content. Basically, I want to discuss on the terminology because we don't want misunderstand the definition of what is food supply chain, what is food security, what is agriculture resilience, okay? Then I will look into the FAO, Food and Agriculture Organization definition and how they're dealing with the issue of food security, okay? And I will go to discuss on the Malaysian agriculture sector at a glance, okay? Our rice plant, okay? Of course, my focus will be on the paddy rice plant, okay? And I will discuss on the impact of pandemic in this rice plant industry as well, and what are the corrective measurements being taken by our Malaysian government, mainly Ministry of Agriculture and Food Industry. Okay, before that, because seriously, we don't know to whom I'm talking. So I have a small activity for all of you, including ibu-ibu, bapa-bapa dekat Indonesia. Tolong saya nak berkenalan dengan kamu semua kerana kita dekat dunia maya sekarang, visual maya kan. So harap kamu semua boleh dengar saya. Okay, boleh tak scan QR code ini ataupun ambil link ini. Okay. Letak your selfie picture very fast, very quick. You only need a few seconds to do that. Okay. Jom kita berkenalan. I want to know with whom I'm talking actually. Okay. Can you do that? From UPN? UPN, can you hear me? Salma, boleh dengar kan?
Boleh, Doktor.
Boleh, okay. As I said, the terminology food security ataupun dalam bahasa Melayu kita panggil sebagai kawalan makanan. Maksudnya adalah to ensure everyone in population have accurate, sufficient enough food untuk makan. Okay, itu definisi untuk food security. Food availability maksudnya adalah kita boleh ada domestic available food and kita boleh import food daripada overseas pun boleh. So itu maksudnya food availability. Even though kita dapat food bantuan, itu pun dipanggil sebagai food availability. So food availability, satu domestically kita boleh produce, kedua kita boleh import, okay, import kalau tak cukup kita import. Ketiga adalah bantuan, okay, bantuan makanan daripada mana-mana NGOs ke pihak luar, itu dipanggil sebagai food availability. And food supply chain, I assume you all know, okay, from the farm till to the table, okay, who are the players who involved in food supply chain, okay, kita akan that is food supply chain lah. So to managing this supply chain is not easy task, okay, but I have to tell a lot of opportunity along the supply chain, especially on the sector, agriculture sector and food sector. We have a lot of potential business opportunity. That is on terminology on food supply chain. Okay, agriculture resilience. Okay, katakanlah pandemik ini, petani macam mana dia nak pasarkan produk dia, macam mana dia nak tanam. Okay, kalau dia tanam, hasil dia nak jual dekat mana. Okay, dia kena berfikir. dia kena bertindak walaupun keadaan yang stres, katakan climate change ke, ribut ke, macam mana dia akan bertindak balas ke atas sesuatu senario. Itu dipanggil sebagai agriculture resilience. How the farmer react to certain changes. Okay, that is on terminology. Okay, I want to touch on food security. It's not only issues of Malaysia or issues in Indonesia. It's global issue where FAO, Food Agriculture Organization, mentioned that by 2050, okay, we will be facing a hunger issue. Okay, that's why on sustainability development goals yang kedua, kalau adik-adik tahu, I think adik-adik tahu sustainable development goal kita ada 17. Daripada 17 tu yang nombor goal kedua tu adalah zero hunger. Zero hunger where FAO is really moving towards make sure everyone have food, at least a meal a day untuk makan. Okay, so itu dia main agenda now lah. Okay, so when talk about food security, okay, ini adalah hunger map 2020 . Okay, still ada hari ni, untuk masa ni ada insan-insan yang tak dapat makanan dekat dunia ni. Tak makan pun. Ada yang tak dapat makan untuk 1 hari, 2 hari, ada. Tapi kita bersyukur kita ada makanan hari ni. Dekat table saya ada bread. Okay, so bersyukur untuk tu. Tapi keadaan ini bukan sama di ada yang tempat-tempat lain lah. So that is the main issue where they want to make it as a zero hunger. Means no hunger, no one is hunger for food. Okay, so that is the initiative of Federal Food Agriculture Organization lah. Okay, so there is 3 category when we talk about food security ataupun kawalan makanan ini. Okay, satu adalah food security, kedua adalah food security means dia ada food available. Dekat nation tu ada food, tak ada masalah. Semua ada makanan untuk makan. Ada satu lagi kategori moderate food insecurity. Maksudnya food ada tapi tak mestinya apa yang diperlukan oleh badan nutrien badan dia. Okay, mungkin apa yang dia makan itu tak baik untuk kesihatan dia. Tapi makanan ada. Okay, that is moderate food insecurity. And severe food insecurity, ada yang tak ada makanan untuk 1 hari, mungkin more than 1 hari. Okay, that is severe food insecurity. So bila kamu tengok dekat Asia, kita dekat Asia, tengok 3 daripada all issue, all dekat dunia ni, kalau ikut dia punya data yang diberi oleh FAO, okay, 704 million are suffering from food insecurity. Okay, and from that figure, 354 are from Asia. Asia is means Malaysia pun dalam Asia, Indonesia pun dalam Asia, Thailand, Vietnam semua dalam Asia. Tengok almost half. Okay, and in Asia we have to give a serious attention. Okay, serious attention to this issue. Okay, so I want to talk on what is agriculture resilience means. Agriculture resilience means, as I said, farmer need to be get ready. Okay, farmer need to get ready whenever there is a challenges. Okay, yang paling utama adalah yang sekarang lah, pandemic. How to react to this pandemic? The only industry that not only industry lah. Okay, during this pandemic, the industry that really making money is one, health industry, kesihatan, memang kamu semua tahu kot. Lepas tu pharmaceutical industry, okay, and of course food industry. Okay, first food industry lah, kerana kita tak boleh keluar tapi kita perlu makan. Okay, so lot of opportunities available during this period of time, even to have self-sufficiency of produce. Even ramai dah mula bertanam sendiri, okay, kerana dia duduk rumah, dia dah mula bertanam sendiri. Dulu kan zaman-zaman dulu memang macam tu. Kita tanam, kita makan sendiri. Okay, when it's become a commercialization, then inilah trading, import, export. Okay, so zaman-zaman dulu memang kita tanam untuk diri sendiri sahaja. Okay, so whatever situation, climate changes ke, insect punya ancaman ke, daripada serangga ke, ataupun banyak benda lah yang lain-lain. Okay, so these farmers, because they are the main producer, okay, on agriculture raw materials, so they have to react. That is called agricultural resilience. Okay, how fast they want to act towards any changes. Okay, so to build agricultural resilience, we have to prepare our farmers. Okay, we have to prepare. They have to prepare. They have to adapt. They need to be a transformer. Okay, then they have to. Farmer normally they tend to rely on agency, government agency. They have to change their mindset. Mereka kena berubah fikiran mereka menjadi entrepreneur. Itu pasal kita dekat UNISA, not only UNISA Malaysia, we really focus on the entrepreneurial activity. We want them to create job. Many others, okay, whoever unemployed in the industry, okay, we can. To create job, we need to have entrepreneurial mindset. We have to create, innovate new stuff lah. Okay, so I don't want to dwell on this. Okay, so at glance on Malaysian agriculture sector. Okay, semua Arab semua boleh dengar aku cakap ni aja. Boleh dengar clear?
Dengar.
Salma, that side, I don't know that side they can hear me or not. Okay, so due to this pandemic, okay, there is a slight drop on the agriculture sector lah. That is negative 0.2% only. Okay, but slightly due to the pandemic lah. And even rubber sector, you can see downturn of negative 18.6% and negative 7.9%. Ini adalah kerana kita tak boleh bergerak. Okay, so due to that penurunan contribute 16.1%, fishing 11.2%, forestry 5.2%, rubber 2.5%, oil palm. Our main share is from oil palm 37.1%, and other agriculture activities contributions are 27.9%. Okay, these are the rough view. Kalau paddy, kamu boleh tengok lah. Ini yang hijau pekat ni adalah tahun 2020. Prediction, prediction value, not real value. But 2019, ini adalah dia punya actual value lah. Okay, it's not predicted, it's actual value. So for national rubber also, you can see there is a drop. These all issues are due. Okay, kita ada pepper, kita ada cocoa bean, kita ada cow. For we are exporting our ayam lah. Okay, fresh fruit bunches. Some measurement to replace tembakau to industry ginseng and okay, so surprisingly our import, so the balance of trade is positive 20, but okay, per month they roughly will get 1,547. So, tapi tak beza banyak pun. Okay, lelaki dapat RM1,572, manakala perempuan dapat RM1,434. Okay, so ini adalah self-sufficiency ratio. adik-adik boleh tengoklah. Apa-apa yang lebih daripada 100 maksudnya kita cukup untuk domestic punya consumption. Okay, cukup. So yang kurang daripada 100 maksudnya dia tak cukup. Bila tak cukup kita tend to import. Itu senario. Itu pasal self-sufficiency ratio, some will use self-sufficiency level is very important for us to make sure where we are standing. So berapa tak cukup? Okay, kalau tak cukup, okay, kita ambil daripada kita punya neighbor country, Thailand ke, Indonesia ke, Vietnam ke, okay, untuk cukupkan, okay. So ini adalah dari kategori ikan lah, okay. Kalau ikan yang tak cukup adalah tilapia, ketam pun tak cukup, mackerel pun tak cukup. Yang cukupnya adalah udang, tuna dengan cutlet fish daripada data ni, based on this data. Kalau adik-adik tengok yang livestock, hanya chicken dengan duck egg saja yang cukup. Okey, yang lain semua tak cukup. Maksudnya kita import. Maksudnya untuk cukupkan untuk domestic consumption kita perlu import. Okey. Ini adalah per capital consumption. Okey, maksudnya 1 tahun Nalini consume, ni data macam mana adik-adik nak perlu baca data ni adalah Nalini consume 22.7 kilogram coconut. Okey, contohnya okey vegetable Nalini makan tomato sebanyak 3.6 kilogram setahun. Okey, cucumber 2.8 setahun. Okey, ini adalah secara purata lah. Okay, and on livestock also lah. Okay. Then ini dependency. Okay, dependency of Malaysia. Okay. Boleh adik-adik semua boleh nampak kebanyakannya kita memang depend from outside country. Okay, because of increased population. One of the reason is increased population, urbanization. Okay, people shift from rural to urban. So a lot of factors involved, and yang salah satunya adalah tak cukup. So kita kena import. Okay. So kalau in term of food consumption, okay, of course as I mentioned earlier kita import kan. So apa yang kita import adalah bread, cereal, fish, milk and butter, imported fruit and vegetables, and beef. Okay, ini adalah I import content item. Okay, ada yang lain tapi ini yang obvious lah. Obviously we are importing. Okay, so now about supply chain, food supply chain. Okay, saya harap adik-adik semua faham lah apa tu food supply chain. Senang aja daripada ladang from the farm. How it will reach your table. Okay, hari ini Nalini ada bread dekat rumah dia. Dari mana bread ini? Okay, bread ini saya buat daripada apa? Tepung kan, gandum kan. Okay, gandum tu dari mana? Okay, so the raw material is from where? Okay, and siapa proses roti ni? Okay, lepas tu roti ni travel dari mana sampai ke saya dekat Terengganu? Okay, saya beli dekat Jerti Giant. Okay, cara-cara pengantaran pengagian, that is called as a food supply chain. Okay, so along the way, as I mentioned earlier, very critical, but yet we still get our food, everything, and a lot of business opportunities available. Okay, and nowadays consumer mindset, they're more on mereka nak check ada kalau Malaysian, because majority are Muslim consumer, of course halal logo is a must. Okay, and normally people will check halal logo, tarikh luput bila. Okay, it's all normal. So all products should place with that kind of information. Okay, so that's why here, if you see this side here, certificate of quality, acceptance, inception, traceability, dari mana source itu, dari mana, origin mana. Okay, so everything. Kalau kamu beli barang, kamu akan nampak yang QR macam bar-bar, apa palang-palang kan. So bila you scan tu semua information sepatutnya adalah that is traceability. Okay. Okay, this is for how people still in Malaysia generally, okay, farmers don't have they are price taker. Okay, maksudnya middle people, orang-orang yang tengah ni yang letakkan harga. Farmer akan terima harga tu. Okay, so kalau sini producer biasanya dia kena spend on raw materials, input, utility cost, labor cost, machinery cost. Okay, so processors, labor cost, utility cost, infrastructure. Apa yang mereka akan belanja. Okay, lepas tu wholesaler pula. Okay, ini semua player supply chain. I'm referring to supply chain, producer, processor, wholesaler, distributor, retailer, and all these costs will be charged by you. You kena bayar. Okay, harga tu kamu yang akan bayar. Okay, this is actually what is going on lah. Okay. Okay, sekarang padi pula. Padi dekat Malaysia ataupun rice plant adalah the third most widely planted crop in Malaysia. Okay, so dekat sini the main season adalah 1st August till 28 and 29 February lah. Okay, that is main season. Off season means during the dry period where they depend on irrigation system. Okay, so dia schedule dia punya jadual adalah 1st March till 31st July of the year. Okay, granary area refer to major irrigation scheme. Kita ada 8 granary area dekat Malaysia. Dia bagikanlah 8 kawasan. Okay, so Mada, Gada, Aida Krian, Aida Barat, Laut Selangor, Aida Pulau Pinang, Aida Seberang Purai, Aida Ketara dengan Aida Kemasin Semarak. Aida Ketara ni dekat sangat dengan rumah saya. Okay, these are the map lah. The granary area. Maksudnya dekat sinilah penanaman padi secara besar-besaran dijalankan. Okay, diamalkan. Okay, dekat Malaysia, kita a survey lah. A survey done by, I get this from one article, KRI, Gazana Research Institute. Source daripada Gazana Research Institute. Okay, so mereka dalam report mereka menyatakan bahawa 2.7 million tons of rice in 2016 was consumed by 67% consumed by people, okay, the population, and only 67% was produced locally. Okay, means that the balance 33% we import. Okay, so kalau kita tengok rice importer, okay, normally rice importer adalah Philippines, Malaysia, Indonesia. Okay, rice exporter adalah Thailand, Cambodia, Vietnam. Okay, nanti kita akan tengok data-data who are the main producer of paddy. Okay, the world main paddy producer adalah China. Ini tahun 2014. Nanti saya akan share yang latest data. Okay, China 28%, India 21%, lain-lain 20%, Indonesia tengok 10%, Bangladesh 7, Vietnam 6, Thailand 4. Okay, tapi kita Malaysia ambil banyak, bukan banyak lah, beras daripada Thailand. Okay, import of rice, tengok import of rice by country. Thailand kita ambil 41% lah, followed by Vietnam, Pakistan, Cambodia, India dan lain-lain. Okay, the variety of paddy yang banyak adalah MR220CL2 50%, okay, ditanam. Yang tadi yang saya cakap yang 8 granary area kan, so kebanyakan tanam variety inilah MR220CL2. Okay, ini current data yang saya ambil daripada Google search lah. Okay, so ini the largest rice producing countries. Okay, China still the number 1, then continue by India and Indonesia. Okay, you all are really contributing a lot for the world consumption, rice consumption. Followed by Bangladesh, Vietnam, Thailand, Burma, Philippines, Japan, and Brazil. Okay, I was delivering my keynote speech to Vietnam, one conference, and the way they are moving, maybe the way they are share their inputs, everything, maybe they will be the 2nd place or 3rd place. Okay, in future. they are planning or when looking at their plan, their strategies, maybe, okay, they are moving forward to be the largest rice producing country lah. Okay. So ini adalah saya tak tahu adik-adik boleh nampak atau ibu bapa boleh nampak atau tidak. Okay. Ini adalah anggaran bilangan petani padi di Malaysia. Okay. Kita tengok 2019 kita ada 192,663 farmers. Keluasan padi adalah 282,201. So hektar lah, okey, kawasan keluasan padi lah, okey. Saya tak tahu dah cukup masa ke apa. Ini adalah kalender yang biasanya all this granary, 8 granary, mereka schedule their farming activities, okay, for planting and harvesting, period of wetland paddy lah, okay. So this information from Ghazana Research Institute. Okay, ini adalah real data. Real data senario dekat Malaysia. Okay, untuk paddy kita ada 9 seed supplier. 9 seed supplier je yang supply input for paddy. Okay, maksudnya. And kita ada seramai 194,931 farmers, okay, for production. And millers, millers means the processor lah, okay, ni semua supply chain lah. 157 license, yang ada license millers, yang tak ada pun ada lah, tapi yang register is 157. Tapi kita ada satu organisasi saja, iaitu Bernas, yang control everything. Control all these activities, okay, which don't know lah, maybe in future we will have this sekarang macam monopoli, mungkin in future kita akan jadikan dia sebagai oligopoly structure market ke, depend lah, depend on the government. Okay, bila tengok wholesale and retailer, kita ada 1,660 wholesaler pemborong, dan consumer 31 million consumer lah untuk paddy. Okay, of course Malaysia kita ada National Agriculture Policy. Okay, 1, 2, 3. Semua ni polisi-polisi ni adalah untuk membantu petani untuk meningkatkan dia punya apa orang kata produksi dia. Okay, dia punya tanaman, hasil tanaman dia. Okay, so kita ada National Agro Food Policy dan di mana fokus adalah untuk increase the productivity. Okey, sekarang kita ada kita panggil sebagai DAN 2.0 ataupun National Agro Food Policy 2021 hingga 2030. Okey. Sama dia, tapi sekarang dia fokus kepada ekonomi kerana 3 sustainable dimension adalah ekonomi, sosial dengan environmental. So now this DAN 2.0 yang current one, yang baru out in plate, memang fokus on this 3 element, and of course all the focus on the agriculture is given to improve untuk menjaga kebajikan petani, meningkatkan taraf hidup petani, menambah pendapatan petani, selain daripada itu meningkatkan hasil pertanian. Okey. Dia punya root itu adalah itu, akar dia adalah itulah. Okay. So okay, touch on pandemic. Of course, pandemic start with the health crisis. Okay, so ramai yang meninggal, and we pray for the soul. Okay, dia mulanya kesihatan, isu kesihatan. Tapi efeknya adalah kepada ekonomi, environment, sosial. Yang paling efek turu adalah ekonomi dengan sosial. Okay, sosial di mana recently dekat Malaysia dalam 2 tahun, dalam lingkungan 2 tahun ni 72,000 kes perceraian dilaporkan. Okay, that is on the social category lah. Ekonomi memanglah disebabkan kita dah tutup, banyak orang tak boleh, kesannya adalah kepada peniaga-peniaga kecil, peniaga-peniaga desa, So majority of them, so somehow it will affect, bagi kesan yang tinggi kepada ekonomi lah. Okay, so don't touch. Okay, ni gambar-gambar je. Okay, ni. Okay, kenapa isunya adalah kerana we already dealing globally with all each nation, okay, and trade liberalization is there, interconnected. Okay, so the supply chain is really interconnected. Okay, goods and services have to pass through more stages. Okay, dia kena melalui. Walaupun saya tunjukkan satu line aja di mana daripada farmer kepada consumer, tapi banyak orang, okay, dan networking dia punya, kita panggil dia sebagai spider web, dia punya sarang tu adalah terlalu complicated and is interconnected. Okay, somehow if one not functioning, definitely it will give impact kepada yang lain. Okay. So, of course, the national lockdown and movement of order memang ada efek, tapi efek dia dekat agriculture, disebabkan agriculture sector ni adalah capital intensive. Kita guna banyak machinery, dan pada masa yang sama kita guna banyak tenaga buruh. Okay, bila you suruh semua duduk dalam rumah, siapa nak mengurus aktiviti pertanian. Okay, so first beginning stage, yes, there is a panic attack, situation panic. That is I'm talking on last year March, around March kan, middle of March, dekat Malaysia kita announce the pandemic 18 March. Okay, kalau tak silap lah. Okay, so 1 month, we have to take 1 month time to adjust everything. But thank God kita tak ada issue. Okay, because of the lockdown everything, of course lah, farm production, food processing, transport and logistics, and of course demand mesti ada kesan. Okay, but with God grace, dekat Malaysia tak adalah isu yang tak ada makanan. Okay, saya tak tahu dekat Indonesia macam mana, but thank God lah, everything government take a corrective action. Okay, and make sure that there is no shortage. Okay, so of course dalam agriculture sector kita mainly depend on foreign labours. Okay, and of course there is impact on food availability and market price. Kenaikan harga memang ada. Okay, and consumer pun during pandemic dia punya cara nak pembelian semua lain dah. Okay, mereka look, because pandemic you cannot go out, cannot move, you only will look for product seperti yang penting, makanan you akan cari. So itu pasal yang kedai-kedai lain macam kasut, baju semua kita tak akan beli. Okay, so memanglah jejaslah perniagaan mereka semua kan. Okay, dekat Malaysia kita memang food kita ni kita import daripada China, India, Thailand, Indonesia dengan New Zealand. Okay, we are depending on the kan shortage itu kita kena import. Okay, untuk cukupkan. Kalau in term of rice production, okay, during this period of time banyak package given by government lah untuk boost the industry, untuk memberi apa suntikan. Okay, so sebanyak 11.2 million was given for our rice, increase the production. Okay, and beside that kita ada PRE Prihatin allocation di mana dia bagi 200,000 setiap district. Okay, dia dapat setiap district means bayangkan 1,810 ada farmers ada district daerah yang dapat. Okay, so quite a lot of suntikan diberi oleh kerajaan. So dekat nationally during this pandemic masih beras kita masih cukup. Okay, hanya 30% beras diimport, itu pun tak ada masalah. Okay, cukup lah. Okay, so pada masa yang sama, of course kerajaan pun memberi dia punya bantuan. Lot of corrective measurement been taken, strategies been placed to improve the supply chain, mainly on paddy lah. Okay, of course, always there is a room of improvement for better future. Okay, so like Malaysia, we are totally depend on foreign workers in the agriculture industry. Okay, and in paddy, we need new variety. Okay, to be introduced. So with better yield. Okay, dapat yang hasil yang banyak. And of course, walaupun package stimulus, okay, untuk nak bagi dorongan kepada petani kita, memang ada package-package, tapi perlu lagi suntikan, okay, and irrigation system need to be upgraded in our paddy field, okay, and we need to digitalize, okay, sekarang kita kerana semua talking about IR 4.0, so we have to digitalize the supply chain actually. And we need to have a well-designed, integrated supply chain and out-based subsidy. Okay, subsidi hanya diberi kepada sesiapa yang really perform. Okay, so subsidy should be like that. Okay, and koperasi petani-petani kena dikuatkan kerana as tadi saya cakap, harga ditentukan oleh orang tengah, bukan farmer. Kerana kuasa farmer tu kecil. Kalau dia ada koperasi, senang untuk dia tawar-menawar untuk harga. Okay, and of course we need public and private partnership. Okay, to complement each other, to have a fruitful agriculture and food supply chain establishment. Okay. So ni reference yang saya guna, saya tak tahu ada ibu-ibu bapa dekat Indonesia dengar atau tidak, adik-adik semua dengar. Okay, I wish, thank you very much and I already done with my presentation and thank you Universitas Pembangunan Nasional Veteran Jawa Timur untuk memberi peluang untuk saya hari ini untuk berucap, harap sampai maklumat berfaedah. Okay, and saya mengucapkan jutaan terima kasih kepada fakulti saya Bioresources and Food Industry UNIZA to give me this privilege to deliver this speech. Thank you. Saya done dah. Saya nak tanya soalan tapi tak tahu boleh dengar atau tidak.
Mohon maaf Dr. Nalini, saya terlupa untuk baca CV doktor ni tadi.
Apa? Tak dengar.
Yeah, CV daripada Dr. Nalini. So Dr. Nalini, she is a lecturer from School of Agriculture Science and Biotechnology. She's graduate Bachelor of Science Agribusiness in UPN. Then she achieved a Master of Science Agribusiness UPN in 2001 and Doctor of Philosophy in Agribusiness Marketing UPN Malaysia 2010. Dr. Nalini ni satu alumni dengan saya. Saya pun daripada UPN dulu doktor. Tapi dekat forestry.
Ya ke? Oh, okey. Tahun bila?
Forestry UPM dulu. 2019.
Oh, muda lagi. Aku tua dah.
Postgraduate Diploma in Entrepreneurship, Cambridge 2013. So she is teaching on agribusiness, agribusiness marketing, contract farming, and supply chain management. Every all participant, if you have any question, maybe we have Q&A session to Dr. Nalini, and the next speaker from the okay, thank you, Dr.
Yes, thank you. Thank you, Mas Fahri, moderator. Saya minta tolong operator, yes, Mas Iqbal untuk melanjutkan tadi artikel saya. Yes, in video. Yeah. Bismillahirrahmanirrahim. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Rabbi shrah li sadri wa yassir li amri wa hlul 'uqdatam min lisani yafqahu qawli. Alhamdulillah, we meet in global. I share my article. my team, I am and Dr. Nora Agustin and Dr. Ramdan Hidayat. Implementation auxin, application and plan at a number of root and kecibiling by Suryanti. This is roots which are not given IPA, E and PS. This is concentration 1 ppm IPA. Implementation auxin, application and plan. This is at IPA. Increase plant with weight and the number of leaf of kecibling. And then implementation gibberellin was spray on all flower panicle. This is expand concentration 5 ppm and the produce seedless spread. This is a parthenocarpy. And then gibberellin, the grape vines on the left is not treated. The plant of the right grow from a grape vine that is sprayed with gibberellin during its fruit development. And then effect of cytokinin stimulate rRNA and protein synthesis. Cytokinin also slow down leaf deterioration in intact plant because of this anti-aging effect. BAP and IAA, formation of kiwi leaf and bud was not obtained in the composition of the media which edits this, which were 4.57 and strand on 1.45 should per expand respectively. And then addition in the composition of the media, the color of the leaf form is brighter. KIPA increase the growth of eucalyptus cutting. And then ethylene activity in ripening fruit will decrease with decreasing temperature. For example, in apple store and 30 derajat Celsius, the use of high concentration of ethylene doesn't have a good effect of the ripening process and respiration. And leaves that fall in autumn, the point where the leaves are released is an abscission layer which is located close to base of the petiole. And this is ethylene on leaf abscission. This combination of red, yellow, and orange pigment that are newly created during the fall in the leaf but appear to change due to the breakdown of the dark green chlorophyll in the fall. Implementation of FCC ASIC for preservation able to survive of the if on the week on media MS with various concentration of abscisic acid and paclobutazole for preservation. The use of paclobutazole can overcome rosette only on the snow queen cultivar. An application paclobutazole caused abnormal culture growth. This is a rosette at the post-storage regeneration state. Studi kasus my research about produksi metabolit sekunder and micropropagation. The product metabolit sekunder, my research about in vitro culture technique of Camellia sinensis to produce epicatechin with phosphor inducer. Leaf Camellia sinensis contain a wide variety of secondary metabolites have potential to support pharmacy, agriculture, and medical as anti-cancer. Indonesian government nationally programmed the new medication study from most recent nature substance by combine the power of plant with modern research result. Cultivar improve in 2.5 and 2.9 by bioactive secondary metabolite production. Secondary metabolite can be obtained from the tea plant that is influenced by required plant waiting time up to several years, weathers, temperature, harvestation, and also cultivation. The purpose of my research, obtain bioactive from culture in vitro Camellia sinensis L product that are relatively faster to obtain than can be applied to several areas of medicine. The benefit knowing that bioactive secondary metabolite has the potential to support health. The risk method, sterilization, process of making stock solution, media production by adding growth hormone, callus with phosphor inducer, cell suspension induction, qualitative identification, and test efficacy bioactive secondary metabolite. In this case, efficacy based on literature study. This is method about sterilization, and this is method about stock solution. This is method about callus bioactive secondary metabolite induction. This is research method about cell suspension catechin secondary metabolite induction. This is isolation and identification callus suspension bioactive secondary metabolite. And then this is efficacy about bioactive secondary metabolite by Alindania catechin for reduce diabetes. And then test the efficacy bioactive catechin for green tea to prostate cancer prevention. And then by injected flavantriol can make highly dynamic in natural soil. And one catechin compound can be allelopathic effect. This is my research result about callus, wick, and extracted, and then analysis qualitative with microscope fluorescent, and then my research about analysis with NMR, and then my research about analysis with SPLC, and then my research about alkaloid with SPLC. This is efficacy to reduce obesity. This is peroxisome proliferator activated receptor identification. Yes. Conclusion my research. This is bioactive secondary metabolite from Camellia sinensis that can be harvested in relatively short time of about 1 month after the induction culture. Bioactive epicatechin found in Camellia sinensis plant that picked from the field can also be produced through in vitro culture technique and obtain epicatechin of 1.7%. This acquisition is related to the administration of phosphor industry. My research funded by the Ristek Dikti (2.17 till 2.19), and the study. This is micropropagation, the research method, callus with phosphor, and then initiation, multiplication, rooting, and acclimatization. This is reference, and this is curriculum vitae. This is personal identity, and then research experience, and community service experience. This is my publication, and then this is oral presentation, and alhamdulillah, this is my patent. I have a award from government and association of other institution. This is okay. Thank you. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh. Mohon maaf bila ada kekurangan. Mudah-mudahan kita bisa berkolaborasi dengan baik. Assalamualaikum.
To the Dr. Sutini and Dr. Nalini for interesting topic. For our student, maybe you can boleh pakai bahasa Indonesia ataupun bahasa Inggris ya. Silakan menulis di kolom komentar ataupun langsung angkat tangan dan berbicara secara langsung. Jadi make it easy ya, boleh bahasa Indonesia ataupun bahasa Inggris. Silakan, apa ada yang tanya? Mungkinnya online.
Padi memang kurang sikit kan. Dia sebab soil fertility dekat Malaysia ni memang acid kan. Itu macam mana padi tu boleh tumbuh dengan baik dekat sana?
Oh, itu soalan. Oh, itu teknikal pula. Bukan kurang, kita tak cukup. Kita tak cukup untuk population. Apa yang kita hasilkan tu tak cukup. And banyak macam tadi saya kata ada 8 granary area kan, Mada, Kada, Aida. Okey, tempat-tempat yang dipilih itu memang sesuai untuk penanaman padi. Okey, padi perlukan air, pengaliran air yang bagus juga untuk pertumbuhan dia. Okey, sekarang kita punya masalah not quality of tanah, dia tanah tak cukup. Okey, kerana tanah kita banyak digunakan untuk industrial crop. Kalau adik tahu kan, kelapa sawit memang terlalu banyak tanah diambil untuk kelapa sawit. Dengan rubber. Okey, so padi adalah tanaman yang ketiga. Dalam presentation saya pun dah cakap 3. So banyak isu. Tapi memang 88 granary area itu memang sekarang kalau mereka punya strategi adalah mereka nak tukar variety so boleh dapat banyak hasil dia. salah satu strategi mereka nak tukar variety. Mungkin kawan-kawan saya yang lebih baik kot boleh tambah kalau saya silap cakap lah. Tukar variety, lepas tu macam ketara yang tempat yang saya ada ni, dia nak pastikan dalam 1 hektar tu dia boleh hasilkan 10 tan padi hasil dia. Tapi setakat hari ini mereka boleh hasilkan 6 tan sahaja. Itu pasal mereka nak tukar variety. Mungkin disebabkan oleh variety dia tak dapat yield yang banyak. So isu sebab kelapa sawit diberi keutamaan sini. Industrial crop. Itu apa yang saya faham lah. Adik dengar?
Dengar doktor.
Okey.
Saya memang tengok rubber, lepas tu sawit lagi banyak betul.
Sawit banyak. Pergi mana-mana pun sawit banyak.
Lepas
And
tu,
you all our competitor actually, pesaing.
apa doktor?
Indonesia pesaing kita.
Oh gila. Tapi memang sana
Industri kelapa sawit kan?
karakteristik padi memang beza sikit daripada sini. Kalau dekat Indonesia dia lekat kan, kalau dekat Malaysia dia tak lekat beras tu.
Okay.
Jadi if you going to Malaysia, the rice is different from Indonesia.
Dia punya variety and the quality
Dia lagi tak lekat. Kalau Indonesia lekat. Okay, all participant. Doctor, I have any question from Hanif Basor. Negara Malaysia kan memang famous sebagai negara penghasil sawit dalam skala besar. Menurut doktor, macam mana negara Malaysia ni boleh memanage kebutuhan pangan di Malaysia yang berupa padi dengan industri cash crop kelapa sawit yang memberikan keuntungan besar?
Bagaimana negara Malaysia
Malaysia memang dia macam mana dia boleh memanage kebutuhan pangan dekat Malaysia, kebutuhan makanan. Sebab dekat Malaysia ni memang
kebutuhan tu maksud apa? Kebutuhan tu apa maksud?
Oh, tak. Needed. Need.
Need. Okay. So macam mana, boleh tak saya faham soalan dulu? Saya tak faham ni.
Alright. Kolom chat ada doktor?
Apa?
Dekat kolom chat.
Kolom chat ada, tapi sekarang masalah saya untuk nak faham soalan tu. Okey, menurut ibu, bagaimana negara Malaysia bisa memanagement kebutuhan pangan?
Needed.
Kebutuhan pangan tu apa? Management aku tahu, mengurus.
Need consumption.
Consumption. Okey, padi.
Berupa padi dengan industri cash crop kelapa sawit yang memberikan keuntungan besar. High benefit.
That's why the issues here during our Tun M time, Tun Mahathir, you focus on industrialization. So what happened? They give more focus on industrial crop rather than food crop. That is the issue now. That's why our import bill, kalau tengok, kita bergantung kepada Thailand, Vietnam untuk cukupkan makanan kita dekat sini. Inilah cara untuk mengurus. Kalau tak, memang tak cukup lah. Itu pasal kita ambil daripada Thailand, beras lah. Thailand, kalau tengok data tadi kan, daripada Vietnam pun ada, Cambodia ada. Okay, so import tu untuk mencukupkan apa yang kurang sini. Macam mana mereka mengurus lah. Saya tak tahu saya faham soalan ni atau tidak. Kawan-kawan saya boleh tolong kot. I don't know, I really understand or not. Because kelapa sawit, kenapa diberi tumpuan kepada industrial crop? Kerana pulangan dia. Itu je. Pulangan daripada paddy. If you compare kelapa sawit dengan paddy, of course pulangan kelapa sawit adalah banyak. So kalau saya farmer, saya akan buat yang mana satu? Kalau katakan sendiri saya, adik farmer petani. Adik mesti tanam something yang akan bagi pulangan yang banyak kan? I don't know lah, I didn't answer or not.
It's okay, doctor.
Okay.
For second question, apakah negeri dekat Sabah dan Sarawak punya lahan padi?
Dia ada tanam padi.
Sebab dekat Borneo tu dia ada, kat Kalimantan Tengah dia ada lahan padi. Lepas tu macam mana dekat Sabah ke dekat Sarawak, apakah dia ada tak?
Mereka ada penanaman padi, tapi bukan skala besar macam apa yang di dekat Kalimantan lah. Padi ada dua jenis kan, satu dia tanah rendah, satu padi umah. Padi umah mahal lagi lah, padi di bukit tu.
Oh, okay.
Padi bario, they call it as a bario rice. Harga memang tinggi. So they more focus on the rice, specialty rice. So bila fokus tu nilai rice tu tinggi lah, tapi ada penanaman padi dekat sana ada.
Oh.
Tapi compare dengan Kalimantan banyak lah.
Dekat mana doktor? Dekat Sarawak dan Sabah tu? Kat
Oh,
mana?
kawasan.
Kawasan kat mana?
Oh, tak. Saya takut aku bagi data salah pula. I kena refer. I need to refer. sorry.
Okay. Oh, dekat Ranau.
Ranau. Okay. Someone is okay. Thank you.
Belum
Betul Sarawak. Ni.
Belum datang lagi dekat Sarawak. Dekat Sarawak.
Oh, dekat je kot.
Peninsula je dah habis. Dah mana-mana dah pergi. Okay,
Mr. Almi tu orang Sarawak.
Oh,
daripada Uniza tu yang jawab betul Sarawak. Okay. Thank you Almi.
Okay, lainnya apakah ada pertanyaan dari mahasiswa Indonesia ataupun daripada Uniza?
Masih with UPM are you?
Lah doktor, alumni dekat sana.
Alumni UPM. Okay.
Silakan, ada yang ditanyakan lagi? Atau mungkin ke Dr. ini? Yes, thank you. Ya Mbak.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Intan Febriana Samastro dari UPN Veteran Jawa Timur. Saya akan bertanya kepada Ibu Sutini. Begini Ibu pertanyaannya. Apakah auksin dengan sitopenia itu digunakan secara bersamaan itu dapat mempengaruhi perkembangan suatu tanaman? Karena kan kalau auksin itu menunjang pertumbuhan akar, tetapi menghambat pertumbuhan tunas dan juga penghambatan bunga dan tumbuhan. Tetapi kalau sitopenia kan untuk mensupport pertumbuhan tunas. Begitu Ibu pertanyaannya. Terima kasih.
Yang lain? Bagus Intan pertanyaannya.
Perkenalkan, nama saya David Christian dari UPN Veteran Jawa Timur. Ingin bertanya ke Ibu Dr. Nalini. Berdasarkan poin SDGs, Sustainable Development Goals, at number 2, bagaimana peran petani, padi terutama, untuk menyikapi climate change atau perubahan iklim, padahal dunia juga dituntut untuk menjaga ketahanan pangan, terutama di negara Malaysia. Terima kasih.
Terima kasih Mbak Intan Febriani. Ya, kemarin dari kelas mana ya? Kelas C, oke. C3. Oke, kita sambung lagi ya. Jadi begini, untuk ZPT itu ada yang bekerjanya itu mandiri ya, ada yang dia kerjanya bersinergi. Nah, seperti auksin dan sitokinin itu memang saling berseberangan sebetulnya kalau dipandang dari segi teorinya. Namun dia kalau sudah diaplikasikan pada suatu tanaman, itu pun kalau kita ambil misalkan yang gampang itu contohnya di in vitro ya. Kalau di in vitro itu kalau di-combine itu malah saling bersinergi. Nah, disinilah menariknya, makanya selalu saya tekankan, saudara itu jangan takut salah ya. Karena dalam suatu tanaman, satu tanaman dengan varietas yang sama, kemudian diambil dari eksplan atau jaringan yang berbeda, itu cara menangkap ZPT-nya itu akan berbeda. Makanya kemarin kan ditambahkan materi tentang bagaimana kalau sudah diaplikasikan, misalkan disinergikan antara tadi auksin dan sitokinin itu diamati. Misalkan keperluan kita mau ke daunnya, misalkan ke sayur ya, kita cukup daunnya. Nah, nanti kenapa itu kok harus bersinergi? Nah, itu harus kita lihat pada tingkat molekulernya. Makanya kemarin kan Ibu tambahkan tentang bagaimana kita melihat ekspresi gennya. Nah, di situ menariknya ya. Jadi makanya kalau orang tidak mendalami tentang ZPT, itu seolah-olah sejak kemarin kok hanya itu ya. Nah, padahal dia itu tergantung dari site-nya. Ya, jadi gennya misalkan kita mau ke yang daunnya aja ya, bisa jadi gen yang ada di daun dengan gen yang nanti ada di bunga itu berbeda. Makanya kalau kita meneliti tentang ZPT ini tidak ada habisnya. Ya. Jadi yang tahu ya peneliti sendiri, kalau dengan kasat mata susah untuk diamati untuk perannya ZPT. Tapi dia sebetulnya memang ada. Makanya kita harus menggunakan ini yang sekarang lagi tren di COVID itu apa Mbak yang dipakai sekarang? Selain swab itu kita apa? Kalau kita mau pergi pakai pesawat kan, ya PCR. Nah, itu tolong saudara kalau ke puskesmas di tes PCR itu coba tanyakan bagaimana cara kerjanya. Nah, kalau di ZPT ini lebih dari PCR, jadi harus yang pakai yang real time, yang RT-PCR. Nah, kalau kita memang misalkan dari sarana dan prasarana kita itu memang sekarang masih terbatas ya, harap bersabar, tapi insya Allah nanti pasti ada jalan keluarnya. Nah, itu kalau saudara menekuni ZPT, itu saudara paling tidak itu harus melihatnya dari tingkat sel. Ya, kemudian dari tingkat sel ke molekulnya supaya bisa mengamati ekspresinya tadi. Makanya kalau lo kenapa auksin sama sitokinin bersebelang apa ya dari teori ini. Tetapi kalau dia diterapkan dengan kita mengoptimasi konsentrasi, maka yang tadi teori yang sains basic itu ya apa ya, bukannya tidak berlaku ya, tetapi ini lagi ada perubahan lagi karena tadi ada optimasi penggunaan konsentrasinya. Nah, juga misalkan nanti saudara menambahkan jibrilin, ya. Tinggal orientasinya kemana? Kalau orientasinya mau ke pembungaan atau pada biji, ya mungkin memang kita cukup menggunakan jibrilin saja. Namun yang perlu saudara ingat ya, bahwa ZPT yang Ibu kemarin sampaikan ada berapa itu 5 ya sementara ini. Itu tadi tidak sama ekspresinya. Jadi makanya sebelum saudara menggunakan ZPT ini, mesti harus secara alami dari tanaman tersebut itu komposisinya sebetulnya apa. Karena di sana ada namanya ZPT endogen. Gitu. Nah, endogen itu sebetulnya ada, tetapi dia akan ekspresi, akan muncul apabila ada pemicunya, ada indusernya. Nah, nanti kalau induser itu juga macam-macam ya. Ada induser yang ikut dalam metabolisme tanaman tersebut, ada yang kita menginduser dari luar. Nah, seperti kalau kita mau pemupukan saja ya contohnya. Saudara kan paling tidak harus tes dulu. Tadi yang ditanyakan oleh Mas moderator tadi ya, jenis tanahnya. Nah, pada ZPT juga itu, seberapa ZPT endogen yang ada pada tanaman tersebut. Nah, setelah itu tujuan kita penelitiannya kemana? Tuh, ya. Nah, kemananya itulah saudara boleh menggunakan teori-teori basic tentang masing-masing ZPT. Ya, setelah itu baru saudara aplikasikan. Untuk aplikasikan juga setelah tadi dianalisis kan endogennya, ZPT endogen seberapa, nah tujuannya mau kemana, lah studi literatur. Ya, makanya kemarin kan saudara kalau buat makalah nggak cukup dari 1, 2 ya. Misalkan topiknya 1 jurnal, saudara harus mengcombine minimal untuk jadi proposal ya. Itu ada 30 proposal. Itu baru bisa menjawab yang basic. Makin bingung ya Mbak. Tapi alhamdulillah kalau saudara bingung itu saya terima kasih, berarti saudara memperhatikan. Next, insya Allah saudara pasti bisa. Ya, begitu ya Mbak Intan jawaban dari Ibu. Jadi ada endogen, ada ZPT eksogen. Supaya kita bisa menerapkan yang eksogen tadi, ya endogen harus dilihat dulu, diperiksa dulu. Seberapa yang ada pada jaringan ya tingkatnya malah, bukan pada tanaman. Misalkan kita mau stek, ya di daerah stek itu aja dulu yang kita uji. Baru kita, oh ini perlu ditambah auksin aja. Misalkan. Oh saya mau yang tanaman cepat berbuah, berarti kita menggandeng gibberellin. Oh saya pengen tanaman ini sudah berbunga, sudah berbuah, ingin dipercepat pemasakannya, kita coba gandeng ZPT di sini, etilin. Ya, gitu ya. Jadi memang agak kompleks. Tapi kalau saudara saya lihat ini kelas-kelas excellent, insya Allah saudara memang ini yang saudara cari. Ya, yang lain lagi? Atau Mbak Ibu? Ya, ya cukup ya. Terima kasih. Ini saya di moderator.
Okay, question to Dr. Nalini. Dr. Nalini, are you there?
Yeah, I'm here.
Okay, silakan tanya lagi. Udah. Doktor faham tak tadi apa soalan daripada student sini?
Tadi belum jawab lagi ke?
Oh, maybe saya boleh student untuk bagi soalan lagi, Doktor ya?
Boleh.
Silakan.
Harap saya boleh jawab lah.
Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi.
Selamat pagi.
Bagaimana petani di wilayah Malaysia?
Bagaimana farmer dekat Malaysia?
Oke.
Menyikapi perubahan iklim yang terjadi di masa ini.
Dalam menyikapi climate exchange.
Oke.
Kita dituntut untuk menjaga ketahanan pangan. Ya.
Nah, sedangkan kita kena dituntut untuk menyikapi menjaga ketahanan pangan. Resilience.
Ketahanan pangan.
Yes, resilience.
Resilient, okay. Bagaimana farmer menjaga
macam mana farmer
how they are adapting with the climate changes.
Yes.
Oh, okay. Our farmers, okay, basically in Malaysia, 93% of farmers are in the category of small farmers. Okay, most of them in any industry are given subsidies by the government to improve their yield, to face all the challenges. Okay, so that is one of the way that strategies that farmer use to improvise their farming practices. Okay, so ni farmer dekat sini di Malaysia memang berkait rapat dengan agensi-agensi kerajaan. Boleh aku.
Ya, Doktor.
Halo. Okey ke?
Okey.
Tak, kerajaan memang bagi banyak subsidi di agriculture sector ni sebagai suntikan untuk menjaga beberapa issues. Okay, in term of harga input kan sekarang naik, climate changes pun mereka akan change the schedule of planting, harvesting. Okay, so many action been taken, but with the help of government, with the aid of government. Still, because they are in the small category, they cannot cope up with the changes, whatever changes going on. Okay, whereas big farmers different, mereka can stand alone. Mereka tak payah bergantung kepada kerajaan. Disebabkan 93% adalah 90 hingga 93% adalah petani-petani yang kecil. Okey, mereka memang perlukan suntikan dari segi kewangan, input adalah daripada kerajaan untuk memajukan bidang pertanian. I don't know, I did answer or not. I seriously don't know.
Okay.
The resilience is question on resilience.
Do you have any feedback? No? Okay, cukup.
Cukup. Kalau in term of padi, kawan saya bantu ni, kawan saya Dr. Nazwa bantu. In term of padi, kawan Nazwa yang bagi answer ni ya. Variety, different variety, which I already touched earlier itself. We are looking for a better variety to get better yield. Okay, so that are some of the strategies for resilience measurement lah, for resilience measurement.
Alright. Okay. Because we have a time is limit, right?
Okay. I'm sorry if
Okay,
some communication problem. I'm really sorry.
It's okay.
Okay.
Maybe student daripada Malaysia yang dia bertanya kepada Dr. Sutini sebab masa ni sangat terhad. So Dr. Sutini nanti boleh bagi jawapan dekat kolom chat. Okay, in the next second session is about entomology, which delivered by Ramadhani Mahendra Kusuma and Dr. Salma Mohammed. Before we started, I would like to read Mahendra CVs. He is a lecturer in Agroteknologi, Faculty in Agriculture, UPNVJT Jawa Timur. He graduate Bachelor of Agriculture in Plant Protection, Universitas Brawijaya, and MSc in Plant Biotechnology and PUST Taiwan. He is teaching in plant pathology, integrated pest management, and plant molecular biology. Without further ado, I would like to invite Mr. Mahendra to present for 40 minutes with titled Biological Control as one of integrated pest management or IPM component. Mr. Mahendra, time is yours.
Okay, thank you. Is my voice clear enough?
Yeah.
Okay,
Very
thank you.
clear. Yes, I can see clearly.
Okay, thank you. So, as mentioned before by moderator, my name is Mahendra Mahendra Kusuma, and my educational background is in plant protection and plant biotechnology. And my experience from the past several years has been mainly in plant pathology, entomology, and then integrated pest management, also plant molecular biology. So, well, so what exactly is integrated pest management or IPM? You know that PHT ya, pengendalian hama terpadu. Okay, so integrated pest management or IPM is comprehensive approach to pest control that use combined pest management practices or tactics to reduce the status of pests to the tolerable level. Well, and also it's based on ecological method, so it's maintaining environment quality. So that's a really kind of in-depth definition. So what is IPM concept? Well, it integrates multiple pest management tactics, just like in the slide: chemical control, biological control, cultural, and also mechanical control. It integrates management of multiple pests, so that includes insects, weed, disease, pathogen, nematodes, vertebrates, and many others. It does not emphasize pest eradication or elimination. So kita tidak menghilangkan, we do not eliminate the pest, but we just control it to the tolerable level. So one goal is to have a rational use of pesticides. We want to reduce environmental contamination and costs, so involving our soil, groundwater, surface water, pollinators, wildlife, and other endangered species. So we want to utilize the natural biological controls to converse and augment, to use selective pesticides and a proper timing of those applications. And then obviously, we want to minimize pesticide resistant problem. As we know, resistance has become a problem in a global world, right? It's because of the use of pesticides. So we want to minimize the pest resurgence also, and secondary pest outbreaks often caused by the elimination of natural enemies with pesticides. So list here in the slide 4 key steps to an IPM program. And these are things we should know. One is know our pests and the plant ecosystem. We want to be able to decide what is an unacceptable pest damage for your situations. Then we want to consider all the available pest management practices. And then lastly, you want to time pest control with windows of opportunity. So identifying these windows of opportunity, all types of pests have a life cycle or set of developmental events that occur during their lifetime. The type of life cycles will vary with the pest. However, most pests have certain weak points or windows of opportunities during their life cycle, when they are the most easy to control. As we can see, for insects, these windows are often during the immature stages. Control tactics will be targeted for this weak point. So you can see in the diagram there, we have the standard insect life cycle with the adults, the eggs, the immature stage, and then the pupa. So a pupa, we can usually target that stage because it's in the ground or hidden in certain place. And sometimes adults or imago are tricky because they are highly mobile. They fly away, easy to fly away. So usually we want to try to get the immature stage, when we are controlling pests. So in the larva stage usually. Then, so let's start with this cultural control practices. We have land and water management. In some situations, water level can be regulated to reduce pest problem. So this can involve mosquitoes or aquatic weeds. And then next is soil tillage. This can disrupt any overwintering life stage of an insect or disease pest. And then weeding. Removing weeds around our production site is a good idea because weeds often serve as alternate hosts for insect and disease. Then sanitation or just garden cleanup. If you can remove, prune, gather plant parts and debris that serve as a protective or overwintering site for many pests. So that will be a good idea. Then we have habitat diversification. The habitat may subsequently increase the diversity of animal life in that habitat because such diversification can attract beneficial or natural enemies, musuh alami, or pest natural enemies. So you have the healthy balanced ecosystem. And then crop rotation. This makes it challenging for pests to access their target host if they are relocated every season. And then tolerant or resistant species and cultivars. So we have to use insect and disease resistant species of plants whenever they're available. And then lastly, we want to focus on the nutrition of our soil, as mentioned before by moderator Mr. Fahri. So also, you cannot change the soil type in your area. There are some practices you can follow to improve the growing conditions for your plants, such as a proper fertilization. But over-fertilization can lead also to excessive lush growth of the plant. It can also be attractive to aphids and other foliar pests. Increasing organic matter of the soil where it is low can improve the growth and health of our plant overall as well. So next we have our mechanical control practices. Hand removal, so when we pull out weeds or other pests, we are physically removing them from the plant. And that's obviously probably the #1 form of pest control. And mowing, if you mow down weeds, especially before they start to produce seed, this can be a very effective pest management tool for the long term. Also, physical barriers. There are many types of physical barriers that can be used to block and disrupt the movement of the pest, just like have a row cover, just like in this picture. And then the traps. So attractive traps can be used to mask trap and remove pests from the environment or to monitor their activities. So control action can be appropriate timed, and traps are primarily used for insects and vertebrate pests. So attractive traps typically use visual. So like these yellow traps, yellow sticky traps, or odor or pheromones like food bait. So like these slug traps, you can see there. And also passive traps can be placed in area common with pest activity to catch the pests. It's usually for flies. You can use the yellow sticky traps. So next we have chemical control. So this includes our chemicals, pesticides. We define pesticide as any substance applied to control insects, fungi, bacteria, weeds, vertebrate, and other pests. So since the advent of synthetic pesticides, pest managers have become very reliant on their generally it's simple to use and then fast acting and effective attribute to manage the majority of pest problems. But however, in recent times, the resistance of pests to pesticides has become high because they have continually used them. So to reduce the resilience of pesticides as a single approach to pest management, so become familiar with those other forms we talk about, such as biological control, mechanical controls, and cultural controls. So although pesticides have been perhaps overused in recent decades, they're still an important tool within our IPM toolbox. So the avoidance of overuse will allow them to remain a viable pest control option, but if it's for the last option only. Then lastly, we have a biological control that we will discuss more about. So we define biological control as any activity of one species that can reduce the adverse effect of another. So for example, we have predators. This is an organism that eats or consumes another. So this can be like predatory insect or predatory mites, or even like reptiles or birds that eat insects. And we also have parasites. This is an organism that lives in our another and kill it. So we will call that a parasitoid while it's completing its life cycle. So here we have samurai wasp, Trisolcus japonicus, parasitic sting bug egg, a native pest of Asia, we know. Then also we have pathogens. These are microbial agents that attack and infect other organisms. So this can be like a bacteria, a virus, or a nematode. And then we have also sometimes there is herbivorous insects that eat wheat, so which is pretty cool. So an example I had is the bent wheat mite, Aceria malherbe. However, most biological control occurs without the assistance of people. Many predators and parasites and also pathogens occur naturally and are continually working to keep nature in balance. The importance of natural enemies is often not appreciated until a broad-spectrum pesticide, which can kill many beneficial insects as well as the target pest, is applied and a new pest is suddenly released from biological control. It's because become a serious problem. So there are things you can do to encourage the activities of biological control agent already present in your environment. So the main thing is to avoid the use of broad spectrum pesticides unless necessary, and then use selective pesticides and target them to pest problem spots. So we do not use pesticide in all of the field. So we need to monitoring the field and also use it just on the right spot, only on the spot that have high attack of the pests. All right. So one of the potential of using biological control are what we are called non-target effect. So what's very important when we apply biocontrol agents is that it has good specificity for host. And that is really only going to attack the pests that we are interested to control. So entomophagus arthropods may be predatory or parasitic. So most predators are either other insect or arachnid, particularly spider and mites. Predatory mites are important in regulating population of Pithepagus mite, including the spider mites. Some mites that parasitize immature and adult insect or feed on insect egg are potentially useful control agents for certain scale insect, grasshopper, and stored product pests. And spiders are diverse and efficient predator with much greater impact on insect populations than mites, particularly in tropical ecosystem. So the role of spider may be enhanced in IPM by preservation of existing populations or habitat manipulation for their benefit. But their lack of feeding specificity is restrictive. Predator beetles like ladybugs and lacewings have been used successfully in biological control of agricultural pests, but many predator species eat a diverse amount of insects and inappropriate for targeting particular pest insect. So entomophagus insects predator may feed on several or all stages from egg to adult or imago of their prey. And each predator usually consumes several individual prey organisms during its life. Okay, the other major type of entomophagus insect is parasitic, as a larva and free living as an adult. The larva develops as an endoparasite within its insect host or externally as an ectoparasite. In both cases, the host is consumed and killed by the time that the fully fed larva pupates in or near the remains of the host. Such insects, called parasitoids, all are homometabolous insects and most are wasps or flies. Some parasitized eggs like Trichogramma, as you can see in the slide, many of the ecto and endoparasitoid is Apellinidae and Echirtidae. Parasite is mealybug. Scale insect, aphids, and whiteflies. Braconids and Ecernoids have a narrow house specificity. We have pathogens like microorganisms, including bacteria, fungi, virus, also and nematodes. Some are insect pathogens, usually killing their host, and of this, many are host-specific to a particular insect. Some disease organisms are fairly host-specific, for example, a virus, whereas others, such as fungal and nematode species, often have wide host range, but possess different strains that vary in their host adaptation. Thus, when formulated as a stable microbial insecticide, different species or strains can be used to kill pest species with little or no harm to non-target insect. In addition to virulence for the target species, other advantages include their compatibility with other control methods, safety of their use, stopping insect feeding quickly, and its ability to self-replicate. So this usually we call biopesticides. All right, so I only just want to comment a few minutes on biopesticides in terms of this whole biological control idea. So there are dozens and dozens of different biopesticides, and what they are is they are made from other living organisms. So some of this name you might recognize: Bacillus, Beauveria, Metarhizium, Trichoderma. Also, BT. Maybe we can familiar with BT, Bacillus thuringiensis. On the top left, that is the most common. There is actually several different species of Bacillus that treat not only insect but also fungal disease as well. But it works so well because it stored great long periods of time, and it's effective in different temperature as well. So nematodes for family include potentially useful control agent for insects. So the most common commercially are species of Steinernema and Heterorhabditis. They are small soil-dwelling nematodes, and then they associated with symbiotic gut bacteria that are pathogenic to the host insect. They kill their host within 2 days of infection. They can be mass-produced easily and cheap and applied with conventional equipment. They control mainly soil-dwelling beetles and Lepidoptera, but can control some plant-boring insects as well. The insect become flaccid and discolored, as you can see at the picture. They become discolored and dwelling and die. So fungi are the most common disease organism in insects, with approximately around 7,500 species known to insect arthropods. Also, only a few dozen naturally infect agriculturally and medically important insects. At least 12 species of entomopathogenic fungi have been used to develop more than around 170 pest control products. Fungal spores that contact and adhere to an insect, germinate and send out HIFA. They penetrate the cuticle of the insect, then they infect inside the hemosole and cause death for the insect, either rapidly due to release of toxin or more slowly due to a massive HIFA proliferation that disrupt the insect body function. Bacteria rarely cause disease in insects. It's most popular fungi than bacteria actually. But after sapropic bacteria, which marks the real cause of death, frequently infect dead insects. Relatively few bacteria are used for pest control, but several have proved to be useful entomopathogens against particular pests. Bacillus populiae is an obligate pathogen of scarab beetles and causes a milky spore disease. As you can see there, the larva become so milky. They discolor from the black to the white. Is a mass infection inside the insect body. Bacillus populiae is useful in biological control by introduction or inoculation, although it is a bit expensive to produce. Also, the strain of BT, Bacillus thuringiensis, have a broad spectrum of activity against larvae of many species of Lepidoptera, and also Coleoptera and aquatic Diptera, but lack of persistence in the field. BT form spores is containing a protein inclusion called crystal, which is the source of the toxins that cause most larval death. Then virus. Many viruses infect and kill insects, but those with potential for insect control are from just 3 viral groups. Viral species are considered safe because they have been found only in arthropods and unable to replicate in vertebrates. The useful entomopathogenic groups are the nuclear polyhedrosis virus, granulosis virus, both belong baculovirus, the cytoplasmic polyhedrosis virus, and the entomox virus. Baculoviruses replicate within the nuclei of the host cell, whereas the GPVs and EPVs replicate in the host cell cytoplasm. And then they cause larva death and discolor, just like the picture, because they attack the nuclei of the pest. So we will talk about classical biological control, conservation, and augmentation approaches of the biological control. So classical biological control involved the importation and establishment of natural enemies of exotic pests. So this form of biological control is appropriate when insects that spread or are introduced to areas outside the rings become pests, mainly because of the absence of natural enemies. Besides the Vedalia beetle, other successful introductions include wasp parasitoids of the cereal leaf beetle, parasitoids of the alfalfa weevil, carabids for gypsy, moth suppression, and many more. Despite the many beneficial aspects of this control strategy, negative environmental impact can arise, such as agents that fail to control the pest. Then augmentation is sometimes used a general term for the supplementation of existing natural enemies, including a periodic release of those that do not establish permanently. So periodic release may be made regularly during a season so that the natural enemy population is gradually increased to a level where the pest control is very effective. So the difference between classical and augmentation is when augmentation, we just inoculate, we reproduce the natural enemies, then we apply the natural enemies periodically into the field. So they can reproduce time by time, maybe season by season. So the natural enemies population will be increased, and then it can be effectively to control the pest. For example, maybe Trichogramma and Carcia that you can see in the slide are mass reared and released into the greenhouse, where their progeny provide season-long control. So the second one is inundative. Inundative is like we spread mass release of natural enemies. For example, we release Beveria bassiana for pathogen, fungi, and also another streptomyces for actinomyces. So we spread a large mass among natural enemies, but then they can persist in into the field. So this is the difference, inoculation and inundative. And then the last we have conservation. It's like we call a refugia. So it's a broad strategy of biological control actually that aim to protect and or enhance the activities of natural enemies and reduce the effect of pests. So in some ecosystems, this may involve preservation of existing natural enemies through practices that minimize disruption to ecological processes. It can include insecticide use that interfere with predator or parasite. So this may include environmental manipulation by altering the habitat available to improve condition for the growth of natural enemy so that they can grow in that habitat. So we provide shelter, their habitat into the field. So for example, flowering plants such as sesame or can be planted close to the rice crops to provide a nectar source for parasitoid wasps that attack ground plant hoppers. Similarly, the effectiveness of entomopathogens of insect pests sometimes can be improved by altering environmental conditions at the time of applications. such as spraying a crops with water to alleviate the humidity during release of fungal pathogen. So a lot of different biopesticides, and actually this is not for you to read, but just to see. For insecticides, this is not even maybe, I don't know, maybe 20% of the list, but biopesticides companies are registering dozens every year. It is just becoming a booming industry because of the demand of this different type of the product, and they are all for the most organic certified. And then in the summary, gonna be a little bit long, but we'll wrap this up. Biological control is not going to be a silver bullet, but it can be an essential component of an integrated pest management program. So it can be incorporated alongside those chemical controls, cultural and mechanical controls that you are using to limit your pest population. It's definitely not going to replace everything, and it's not going to work in every situation. But sometimes that ever evolving and something that can be typically can be incorporated at least to some extent in most croppings and cropping system, and so specific adaptation or application. So this is, I just want to finish up and give you a resource to get some more information. I mean, there are 100 or different, so sorry. It's different of a biological control, but this is a website that has just about every natural enemy you can think of. So lots of great information here. So if you just Google biological control kernel, then you can come to this website. Okay, maybe that's all for me now. And thank you very much.
Okay, the next speaker about entomology will be delivered by Dr. Salma Mohammed. She is a senior lecturer from School of Agriculture Science and Biotechnology, Faculty of Bioresource and Food Industry, UNIZA. She is graduate bachelor degree on zoology, molecular biology in Universiti Kebangsaan Malaysia in 2000. Then she achieved Master of Science in Zoology in Booth University at 2005. And PhD in entomology in UPN Malaysia in 2018. She is teaching on entomology, integrated pest management, and crop production. Without further ado, I would like to invite Dr. Salma to present for 40 minutes with title Oriental Fruit Fly Bactrocera dorsalis, a major pest of fruit. Dr. Salma, time is yours.
Okay, terima kasih. Thank you to Mr. Chairman, Mas Fari. Everybody can hear me loud and clear?
Yes, I can hear you, Doctor.
Jelas ya?
Dengar.
Okay, Assalamualaikum and very good morning to everyone. Okay, before I start, I share my slides first. You can see the full view of my slide. Nampak ya slide saya?
Ada.
Okay, terima kasih. Okay, kabar semua, saya harap anda semua dalam keadaan baik-baik semuanya. Kepada yang dihormati pihak pengurusan, speaker kita, pensyarah dan pelajar di Universiti Pertahanan Nasional Veteran Jawa Timur dan juga UNIZA. Salam perkenalan daripada saya. Saya Dr. Salma Mohammed daripada Faculty of Bioresource and Food Industry UNIZA. First of all, I would like to thank UPNV Jawa Timur for inviting me to be a guest lecturer today, particularly for entomology. Okay, since we are in the same field in agriculture, So today I would like to share about one of a major pests of fruits known as oriental fruit fly, Bactocera dorsalis. Okay, begin with the taxonomy of this species. Okay, Bactocera dorsalis or oriental fruit fly is a great economic and agricultural important pest due to damage caused to commercial fruit crops. And it is under the order of Diptera, family Tephrichidae. But however, due to some synonymization of morphological, molecular, behavioral, and ecological data of B. dorsalis and its related siblings include the Bactocera invadens, Bactocera papaye, actually in Malaysia, Indonesia, previously known as Bactocera papaye, and also Bactocera philippinensis. So they were considered as the same biological species as Bactocera dorsalis. Okay, in terms of their distribution, okay, as shown in this map, you can see that B. dorsalis distributed all over the world with various host plants. It can form more than 965 countries worldwide. That's why it's very strict quarantine of fruits from those countries. Okay, because as known as oriental fruit fly, due its origin from here, from the East and Southeast Asia, including our country, Malaysia and Indonesia. There are many research conducted about this species for the last 50-60 decades. It's included in Malaysia. If you Google Bactocera in Malaysia, there are many papers or manuscript as well as in Indonesia as well. So this species, this pest, a very important pest since the last 40 years ago. So now come to the host preference. Okay, this particular species actually can attack more than 200 fruits crops, but the most preferred host fruits, including the mango, the guava, papaya, and also star fruits. Okay, now it's come for their its biology. So the question is how we want to know that the species is, how to identify this species. Okay, Bactocera dorsalis has several main characteristics. It is larger than a housefly. Okay, body length is about 8 millimeters. The body color, you can see that from yellow to the dark brown, and on the thorax is black color. The abdomen has two horizontal and longitudinal median black stripes and form a T shape. Okay, and compared to the male, a female has ovipositor, meaning that this ovipositor used to lay eggs. In terms of their life cycle, the Bactocera dorsalis undergo a complete metamorphosis, a holometabolous insect, from eggs laid in cluster and then hatch into larvae. Okay, the larvae, the eggs insert by cluster. Okay, and the larvae come in, form in the 3 instar larvae, and then in about 7 to 14 days, the larvae will form a pupae. And after that, about 10 to 14 days, the pupae will emerge as adults, and the life cycle goes on, and the life cycle took about 1 to 3 months to complete. Okay, for damage and symptom. Okay, these are quite important parts to know how does the fruit has been infested by the fruit flies. Okay, let's see how this pest attack and damage the fruit. Well, it is begun with the female adult, normally searching the mature and ripe fruits. Once the female found the suitable host fruit, she will use her ovipositor is a very sharp and long ovipositor to inject the eggs into the fruits. And then the larvae hatch from the eggs will consume the fruit tissue. And you can see here, all the fruit tissue has been infested by the larvae of the Bacterosalidocellus. Okay, here is I have a footage video to show you how the female of Bacterosalidocellus inserting her eggs into the fruit. Okay, you can see that use is long and sharp like a needle to oviposit eggs under beneath the skin of the fruits. Okay, the first symptom we can see clearly is from the black spot appeared. Okay, the skin is bleached, and then you can see the black spot appeared, and finally the fruit becomes watery, bruised, and rotted or decayed. Okay, also here some pictures show how severe infestation by these fruit flies for the various type of fruits. Here is the larvae inside the fruit, and also inside the guava fruit. When you cut open the fruit, and you can see the damage caused to the fruit. Finally, the damaged fruits will fall to the ground, and then at this time, the larvae, which the lay instar, will crawling from the fruits and jump into the soil to pupate. So the pupal stage form inside the soil. Therefore, without the proper and consistent control, the direct damage to fruits varies from a few percent but can reach up to 100%, depending on the fruit fly population, season, variety, and area. So if we do not control it properly, so the damage or losses can reach up to 100%. So now come to the important question. So how we want to control this pest? Okay, there are many control methods to control Bacterosalidocellus, but of course, integrated pest management of fruit fly is a better approach. Okay, as you can see here, meaning that we not only rely on one single method, but integrate with other methods at the same time. So the combination of different control measures of fruit flies include the physical or cultural, genetically, behavioral, biological control, and also chemical. Okay, now we look into the detail of each control methods. The first one is cultural control, such as food sanitation. Okay, what we can do is collect and destroy all the damaged fruits, and this technique actually to prevent the fruit fly larvae from developing or emerging adult flies, so that they cannot return to the crop to reproduce. The next method we can see is by wrapping the fruits before it reach maturity. Okay, this is a very simple action, but has proven effective at preventing fruit infestation by fruit flies. The wrapping materials can be the newspaper, old newspaper, or paper bags, and this system also provides physical protection from mechanical injuries such as scars and scratches, and in some cases reduce fungal spots on the fruits. Although this type of control method is laborious, but it's much cheaper, safer, and easier to do, and give opportunities to farmers a more reliable estimate of projected harvest. And the best time to wrap the fruit is before it reaches maturity stage, because the pest, this Bacterosalidocellus normally prefers to attack mature and ripe fruits compared to the younger or unripe fruits. Okay, now we look at the next control method using protein bait trap. Okay, what is the protein bait trap? Okay, we know that as female fruit flies need of protein source for full development of her reproduction and also for egg production. Therefore, protein bait trap is the best method to control them or to suppress them. Basically, in the protein in the trap is mixed with some kind of insecticide. So when the female consume the protein bait, it will die. Okay, another approach we can use is by using stainer trap or pheromone trap. Okay, in contrast with the protein bait trap, stainer trap attract only the male fruit flies. Okay, normally methyl eugenol or ME attractant is commonly used. This is actually the standard stainer trap, but actually we can DIY the trap just using a recycled drinking bottle as shown here. All we need just plastic bottle, the wire and the cotton wool or the cotton ball, soldering iron and ME, and then we just make a holes on top or the middle body of the plastic bottle, and then the cotton wool tied to the wire and smeared with ME. Okay, and then the trap just hang on the tree's branch. Okay, just now Mr. Mahendra already mentioned about the parasitoids and predators, and of course for Bacteria dorsalis, biological control using this beneficial insect as biocontrol agent is one of the best method to control the population of the pests, because we know that this biological control agent play an important role in keeping fruit flies control under the acceptable level. Okay, basically these natural enemies are existing in the ecosystem and very known as a farmer's good friend. As you can see here, predators like weaver ant helps to repel female fruit fly from ovipositing eggs into the fruits. Okay, just a little bit information about the parasitoid of Bacteria dorsalis. Okay, parasitoids, of course, the adult females lay eggs on other insects, and basically smaller than their host and defined by its feeding habit of its larvae. And the most important group is from the Hymenoptera, about 75%. And as for parasitoid for Bacteria dorsalis, specifically they are members of Braconid species, member of Opinae subfamily. Also, they are solitary, meaning that they lay only one egg per host. And endoparasitoid complete their development inside the body of their host. And coenobian parasitoid means that they allow host to continue its development until the host is about to either pupate or become an adult. Either the parasitoid is egg or larva or pupa parasitoid, the parasitoid's adult emerge from the host puparium. Okay, this is some example of the parasitoids. Egg parasitoid, either is pupal parasitoid attack the pupa stage, and larva parasitoid attack the larva stage of the host. These are the specific species of parasitoids attacked by dorsalis. It's quite small, about less than 1 centimeter actually. It has a similar characteristic. It has a very long ovipositor because need to use to locate its host under the veneer of skin of the fruits. So now we look at how does the parasitoid search or locate the host. Okay, from this picture, you can see the parasitoid is inserting her eggs into under the skin of the fruits. Actually, under the skin of this fruit, locate the eggs or larva of the Bacterocerra dosalis. Although they cannot see or the eggs or larva inside the fruits, but they have a very sophisticated of their sensory system. Okay. Here is the some footage of video. This is a parasitoids. At the same time, you can see the female of Bacterocerra dosalis also locate the right spot to oviposit her eggs. Okay. Basically, parasitoids foraging behavior rely on several cues, include the visual, chemical, and mechanical stimuli. The cues from the host larvae are mainly vibration or sound created by the larva feeding and crawling inside the fruit. So it can sense the place where the larva or eggs of the Bacterocerra dosalis is located. Okay, now we go to the how to conserve predators and parasitoids or natural enemies in the farm. Because now we know that they are very important to us. So one of the way is to conserve the habitat by increase the flowering plants or beneficial plants in the farm. Because we know that parasitoids require nectar plants, the adult parasitoid require nectar plants to extend its life and also perform as a shelter to protect them from other insects. Therefore, this one of the method is very essential to make sure to sustain the population of beneficial insect in the farm. Okay, there are another control methods that also eco-friendly, known as sterile insect techniques. Okay, this method basically about maze of insect, in this case Bacterocerra dosalis fruit flies, genetically are sterilized through the radiation in laboratory, and then before release in large numbers to mate with the wild females in the wild, leading to the female laying infertile eggs and eradicate the pest population. Okay, maybe you ask why is it safe to us? Are irradiated insect radioactive? So the answer is no. So insects that have been sterilized using the radiation are not radioactive. So it's very safe to us, human, and also the ecosystem. And another way to control is by soil disturbance. As we all know that the pupae of bee dosalis form inside the soil. So by plowing and ground flooding the soil will expose the fruit fly pupae in the soil and increase mortality of the pupae and so on the population of the bee dosalis. And finally, last but not least, using pesticide to eliminate the pest. But of course, in IPM, use it at the lowest level and as a last resort. However, using or replacing them with the biopesticide is a better option because it's a much safer than conventional pesticides. In addition, biopesticide also compounds can be used in insect management system for production of organic fruits or as well as the vegetables. So as a conclusion, controlling bee dosalis fruit flies is essential for better fruit production. And before I end my presentation, I would like to share you here the additional interesting information about bee dosalis. Okay, do you know that ratio of bee dosalis male and female is 1:1, and they are most active time of mating before dawn and before dusk. And this behavior actually is 1:1 is we manipulate them for enhance the control of them by using the pheromone trap or also the protein bait trap. A single female of bee dosalis is capable of laying in between 1,000 to up to 1,000 called 100 eggs over her lifetime. So just imagine that how many eggs per female can produce on her lifetime. So that's why this pest are very rapid in development. And then the eggs are very elongated and elliptical, hatching within 24 to 48 hours. Study shows that these fruit flies can be mass rearing in the laboratory with the right larva artificial diet, either in form of gel, paste, or liquid diet. And normally this mass rearing of fruit flies, the main purpose is for biological control program and as well as for sterile insect technique. This is some example of in laboratory how we rear the insect in the laboratory. The adults actually consume the sugar cube and also the water, and the larva needs the paste or the liquid and also the gel form of the diet. And fruit flies are also reared to produce food products nowadays. We know that many people already turn to protein from the insect. So fruit flies such as B. dorsales is not also include for production of the food for the future protein source. Okay, with that, hope the information I share here will benefit us for better understanding of B. dorsales fruit fly. And with that, that's all from me. And thank you to all the listening for my presentation. Thank you.
All right. I'm sure everyone have a question to the Mr. Mahendra and Dr. Salma for this interesting topic. We would have 20 minutes for discussion, maybe with each 3 questions. Is there any question, please? Okay.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, saya Rita dari UPNVJT Jawa Timur. Saya ingin bertanya kepada Pak Mahendra mengenai dengan pengendalian hama terhadap tanaman. Jika pada suatu lahan itu terjadi ledakan hama secara menyeluruh, maka dapat dikatakan bahwa penyerangan hama tersebut mutlak tanpa kecuali.
Lalu apa langkah pengendalian yang efektif dan tepat untuk mengatasi masalah tersebut? Apakah langkah yang digunakan itu adalah penggunaan pestisida atau masih bisa dengan pengendalian musuh alami? Karena seperti yang kita tahu bahwa pengendalian pestisida kimia itu akan berdampak buruk pada lingkungan dan juga tanamannya. Seperti itu. Terima kasih, Pak.
Okay, next question.
Terima kasih. Perkenalkan nama saya
dari UPNVJT Jawa Timur. Di sini saya ingin bertanya kepada Pak Mahendra juga dengan Ibu dari Malaysia juga ya yang mungkin bisa menanggapi juga terkait pengendalian hama tikus. Karena dirasa hama tikus ini tuh dilakukan beberapa pengendalian tuh masih sulit untuk ditangani. Seperti kemarin pada saat kita dari agroteknologi itu melakukan bina desa di Jombang itu, hama tikus itu sempat dijadikan topik untuk pembahasan bagaimana pengendalian hama tikus yang efisien dan tepat. Dikarenakan jika dilakukan secara kimia itu masih kebal dan juga dengan pengendalian yang lain itu masih melonjak karena tikus ini juga berkembang biak itu sangat cepat. Jadi kalau kita tidak melakukan pengendalian secara tepat dan efisien, takutnya di saat kita sudah menangani hama tikus yang lain, itu malah akan melonjak dengan hama tikus di tempat yang sama, tapi berbeda lokasi seperti itu. Terima kasih.
Okay. Next question, please. Oh, okay. Mr. Mahendra will be answered
Okay, so can you hear me clearly, right? Okay, so how we control a pest? It can be resistant or resurgency. Di atas batas ambang ekonomi, right? In economically threshold di atas, secara menyeluruh. So, yang bisa dilakukan adalah we see what stage of the plant. If the plant is still in the maturing stage or vegetative stage, we can do like mass spreading, like I said before, mass release of the biocontrol if it's a pest. So we can release a parasitoid or predator to the field. It can be an option. So, but if it's like in the fruit, like for example, eggplant that attacked by pomopsis. So I've been experienced that in the farmer in the Pandeglang in the West Java. So the farmers' eggplants get attacked by pomopsis. It's a larger scale. It's almost all 100% of the fields that attacked by pomopsis. So the thing that we can do is to eradicate. We can pull out the plant. But if it's attack all of the fields, we can do no more. Actually, in IPM, we do like a monitoring and preemptive, also preventive. So to prevent is the most effective method approach to control the pest or also disease. So we, the farmer usually gather some groups, they discuss about the right timing and to plant. So they will avoid pest attack of the pest, also the disease in the right time. So the best practice actually to control the pest and disease is the preemptive and preventive. Curative is an option. We can do like spray the pesticide, fungicides, but well then we don't know how fast the disease or a pest the growth in the field. So the best options is actually to prevent. But if it's happened, so the option is we can do a pesticide is an option, but then we have to rotate. Rotasi pesticida. We cannot apply a single pesticides secara periodik ya, periodically. It will because you're right. It will because like environmentally damage into the soil, into the plant toxicity, also from the environmental and the farmer also. So I think the best practice is to control the pest and disease is preventive method. So maybe that's for me. I hope it can answer your questions.
Is there any feedback? Okay. The next question to Dr. Salma. Excuse me, Dr. Salma, saya Hanif daripada UPN, Veteran Jawa Timur. Izin bertanya, penyebaran prosak Bactotera dorsalis menurut PowerPoint Dokter berpunca di sekitaran wilayah Asia dan Afrika. Soalan, mengapa penyebaran sangat minim di wilayah Amerika Tengah dan Amerika Selatan? Apakah prosak tersebut belum sampai dekat sana ataukah tanaman buah kat sana sudah resisten? Dan macam mana pengaruh terhadap budidaya tanaman buah di daerah sana?
Okey, terima kasih Hanif atas soalan yang sangat bagus itu. Okey, berdasarkan slide saya tadi, saya ada nyatakan bahawa Bacillus dorsalis ada Bacillus dorsalis species kompleks. Okey. Under Bacillus dorsalis kompleks ada lagi lebih dari 50 species under that complex species. But there are other species Bactropsera yang memang native ataupun invasif di negara-negara lain. Tapi untuk Bactropsera ni memang kawasan ini tertumpu di kawasan wilayah Asia dan juga Afrika, dan some part of the Amerika Tengah dan Selatan tu memang dah ada kes lah ataupun ada rekod. Kerana seperti yang kita tahu dia mudah tersebar kerana ekspor dan import buah-buahan kan. Sometimes walaupun regulation untuk quarantine sangat ketat, tapi ada juga yang terlepas lah. So dia akan menjadi invasi setempat tu. Boleh juga dikatakan tanaman setengah tempat sudah resisten kepada Bactropsera dorsalis kerana teknologi sekarang kan yang makin maju, penghasilan benih-benih tanaman yang lebih kuatlah menahan daripada serangan perosak dan penyakit ni. Tapi namun kita kena sentiasa aware lah about this pest of fruits kerana dia mudah tersebar secara inilah melalui pengeksportan atau import buah-buahan dari seluruh dunia.
Okay.
Menjawab soalan ke? Harap menjawab soalan lah saudara Hanif.
Gimana Hanif? You have any feedback? Yes, jangan ada. From Amira, from Uniza to Mr. Mahendra, I have two questions for Sharma Ramadhani. What is famous pest recently occurred in Indonesia which give huge loss in crop production? And second question, is there any pest in Indonesia that difficult to handle even to using IPM method?
That's all. Thank you.
Okay, thank you, Amira. It's the next questions. So I'll try to answer as far as I know. In Indonesia, it's the pest that hardly to control is rodent. As mentioned the question before, the rodent is spread out in Indonesia because we have like paddy field, a lot of paddy field here, and also we have like a lot of rodent species here, so too hard to control. Also is handled with IPM. Well, actually in some districts they implemented IPM and it's got successfully, but some of them is not successful. It depends on the approaches that they used and the timing of the method that they use is the right time or not. So, well, rodent is another, and also we have fruit flies, Bactropsera dorsalis, as mentioned by Dr. Salma. So we do have also in the mango and the longan and several crops, fruits of fruit crops is Bactropsera dorsalis, the fruit flies, and it's hard to control also. Maybe that's. I hope this answers your question.
Is there any question again, please? Okay.
Saya ingin bertanyakan kepada Miss Salma tentang teknik serangga mandul atau steril insect teknik, tentang apakah teknik serangga mandul ini dapat dikatakan sebagai pengendalian hama terpadu, karena tujuan dari teknik serangga mandul ini untuk sepengetahuan saya adalah untuk eradikasi total atau pemusnahan total serangga, sedangkan tujuan dari pengendalian terpadu itu hanya untuk menurunkan populasi. Nah, apakah ini juga dalam jangka panjang akan berpengaruh terhadap keberadaan si lalat buah? Dan karena lalat buah ini kan seperti yang dijelaskan tadi bisa bermanfaat untuk food product. Terima kasih.
Mas Farid bisa ulang balik soalannya, kurang dengar.
Jap,
Oh, maaf ya, video saya tiba-tiba ada masalah pula, tak keluar muka saya.
Tak ada masalah, tak ada masalah.
Okay, tapi saya masih boleh dengar lah.
Alright, doktor.
Soalannya.
Saya translate jap doktor.
Okay. Tapi saya ada dengar pasal sterile insect technique, right?
Yes.
Something related to the sterile insect technique.
Yes,
Adakah berkesan untuk mengawal populasi lalat buah? Isn't it?
Betul.
Okay. Okay, seperti yang saya ada share tadi, lalat buah dia punya ratio 1:1. Okay, male to female is 1:1. Macam seolah monogami lah, dia tak poligami. Satu jantan kahwin banyak betina kan. Maknanya jika kita kontrol dia punya populasi male dia ni, sama ada sterile insect technique lah salah satu caranya. So dia akan secara slowly akan mengurangkan populasi lalat buah lah, sebab dia dah dimandulkan tu, dia tak dapat nak lalat buah betina tak boleh nak hasilkan telur yang fertile lah. Itu antara salah satunya lah. Tapi of course kena integrated lah dengan cara-cara yang method lain sekali lah. Harap membantu menjawab soalan saudari tadi.
Okay, is there any feedback? Is that clear? Okay. One question again to Dr. Salma. Okay, wait. About rat. Bisa diulang lagi nggak?
Ada pasal trap ke?
Pasal tikus.
Oh, tikus. Yang soalan pertama ke? Saya pun kurang jelas tadi.
Sekejap doktor, saya pun juga kurang jelas.
Oh, pasal tikus saya dengar trap. Rat.
Tentang bagaimana pengendalian tikus yang efisien dan juga tepat. Kan diketahui bahwa hama tikus itu bukan salah satu hama yang susah untuk dikendalikan secara bertahap ataupun dengan pengendalian yang lain gitu. Itu bagaimana pengendalian hama tikus secara efisien dan tepat. Seperti itu.
Maknanya adakah untuk mengawal populasi tikus di pertanian ke atau tikus yang pest perosak di rumah?
Dekat pertanian. Sebab tikus ni kan memang susah untuk nak dikendalikan.
Betul.
Macam mana nak manage ni tikus
dia
ni?
Membiak pun cepat kan?
Betul.
Salah satu caranya selalunya kan farmers menggunakan racun tikus kan, tapi kita tahulah racun tikus ni ada kesan jangka panjang yang kurang baik. Tapi kalau untuk kontrol secara biologikal, selalunya kita menggunakan farmers menggunakan burung hantu, right, untuk secara kawalan secara biologi. That's why kalau di ladang sawit, even di sawah padi, kita boleh nampaklah burung hantu memang digalakkan untuk populasinya untuk mengawal tikus secara alami lah, selain menggunakan racun perosak yang khas untuk tikus. Tapi sekali lagi ditegaskan, penggunaan racun perosak yang sintetik ni memang kalau boleh paling minimum lah. Dan biopesticide macam Mr. Mahendra share, banyak dah kan orang buat kajian biopesticide, itu lebih baik lah untuk kawalan jangka masa panjang.
Okay, is that clear? Okay, thank you Dr. Salma. One question again please. Ya Mbak. This for the last question.
Assalamualaikum warahmatullahi wabarakatuh. Saya Nisa Zukruf Oktavia dari UPNVJT Jawa Timur. Saya ingin bertanya ke Pak Mahendra. Tadi kan disebutkan pengendalian hama terhadap tanaman itu ada metode biologi, mekanik, sama kimia. Nah, itu yang mau saya tanyakan adalah bagaimana cara kita itu menentukan cara mana yang akan kita lakukan, dan apakah di antara tiga itu ada yang paling baik untuk dilakukan, seperti itu Pak. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Pertanyaannya bagus sekali. Jadi bagaimana kita menentukan metode apa yang digunakan? Jadi which approach method that we can use to control the pest or disease? So actually we have to combine biological control, once again, I said that it's not a single silver bullet. So we need to combine. We have to use either biological control, mechanical control if they're possible, and also chemical control for the last option. If it's the pest or disease is over the economical control, so it causes, how to say, we can predict that it will cause economical damage. So that's only time that we can use pesticides or chemicals. But then when we try to control, we can use either biological and also physical or also cultural practices. We will do, kita menggunakan kultur practice ya, with GAP, good agricultural practices. Jadi bagaimana sanitasi lingkungan, kemudian sistem drainase dan irigasinya bagaimana diatur. Kemudian if it's possible, we can use varietas tahan. Is a resistant variety. Jadi is biological control also. If it's possible, we can use mulsa. So we can combine. We do not. It's not one option actually. We can combine. So it's complex. IPM is complex. So we need to do like monitoring. Monitoring is so important. The thing is the most important thing in IPM is monitoring. So we have to look out into the fields comprehensively, not only just look on window. No, we have to move deep into the fields so we can mapping the land and we can choose whether it's possible to control with biological or it's too high. The attack is too high, so we can choose the optional pesticide. Maybe that's. I hope that's answer your questions.
That clear? Oh, maybe last question from Uniza, Hadin Azhar. Salam sejahtera. I am Hadin from Uniza. I want to ask Sir Mahendra, in Malaysia, apple snail is one of the famous pests for paddy plantation. Is there any problem caused by apple snail in Indonesia? And if there are cases, how do farmers in Indonesia overcome the problems?
Okay, thank you, Hadin from Indonesia for the question. Actually, we have also snail, but most of the snail is attacked now in paddy and also in another plantation. So the farmer usually use onion and also how to say bawang putih. Yeah, for biological control. It's hard to control actually in here, but I don't know so far about the snail in the farther in paddy plantation and apple. No, I'm not really sure about the problem caused by apple snail. I'm so sorry about this. Maybe I will try to look up for the answers. And in Indonesia, I think the snail is Pomassia canaliculata is in the paddy field. We also have that in Indonesia, but I don't know in another plantation. Maybe that I hope that will answer your question.
Okay, I will close for session crop production and entomology, and our rest agenda has entered a break session for 60 minutes. After that, at 1 o'clock, right? 1 o'clock, we return to main room to continue the next session. Maybe dekat Malaysia, pukul 2 suku, kita boleh join lagi untuk melanjutkan ke next session. Sekarang kita berehat dulu. Terima kasih semuanya. Assalamualaikum warahmatullahi wabarakatuh.
Oke, terima kasih semua. Assalamualaikum.
Dr. Salma.
She is positioned now is the Secretary of Department Agroteknologi, Faculty of Agriculture. She graduated Bachelor in Agriculture Science, Universitas Gadjah Mada in 1991. Then she achieved Master of Agriculture in Agricultural Science in 2002 and PhD in Agricultural Science in both University 2007. She is teaching in plant protection. Without further ado, I would like to invite Dr. Insinyur Sriwiyati Ningsih to present for 40 minutes with titled Microbiology for Food Provision. Dr. Wiwit, time is yours.
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Alhamdulillahirabbil alamin. Thanks God. Today we can gather in this classroom. This is my session. I will deliver about microbiology for food provision. Sorry, tak buka aja ya. Yes. So we have lunch. Now time to talk about food. Okay. Ini nih. You will learn about the role of microorganism as food, as agent of fermentation, as PGPR, and as genetic engineering agent. As food, we know that there are some of edible fungi like Volvariella volvaceae, Auricularia auricula, Pleurotus ostreatus, Agaricus bisporus. I think all of you have eaten them, ya. Jamur merang, jamur kuping, jamur tiram, dan jamur kancing. Are you have question why we can see them, but we call them or we include them in microorganism? Ya, ada yang bertanya, kenapa kita bisa melihat bendanya ini, tapi kok dimasukkan mikroorganisme? Ya. Ya, ada yang bisa menjawab? Please, Mas yang pakai batik oranye, can you answer my question? No? This is classroom ya, jadi bukan saya webinar ya, ini kelas, jadi saya punya kesempatan bertanya kepada Anda. Oke, siapa yang tahu? Can you answer my question? We can see them, but we include them into microorganism. Ayo. Okay. Actually, fungi, when they in the first grow, they are as a hyphae, you know, hypha. Yeah, hyphae is a micro size in a micron. So we include them into microorganism. Okay. Mas, bisa Mas. Okay. The other microorganism that can roll as natural supplement, there are spirulina and chlorella. Yeah. Have you heard about chlorella and spirulina? Yeah. Spirulina and chlorella is single cell protein. Yeah. And some industry have packaged them into a them into a tablet or capsule. Okay. The other role of microorganism is agent of fermentation. What is fermentation? Fermentation is a metabolic process that produces chemical change in organic substrate through the action of enzymes. In biochemistry, it's narrowly defined as the extraction of energy from carbohydrate in the absence of oxygen. Jadi dalam proses fermentasi tidak diperlukan oksigen. Kenapa? Ya? Ya.
Ya, karena kan fermentasi kan niatnya untuk memunculkan mikroorganisme. Kalau dapat oksigen kan pertumbuhannya kurang maksimal.
Bukan. Jadi fermentasi bukan memunculkan mikroorganisme, tetapi fermentasi itu memang dilakukan oleh si mikroorganismenya. Tetapi mikroorganisme yang bisa fermentasi ini biasanya adalah yang memang yang non aerobik, berarti tidak membutuhkan oksigen. Ya, biasanya adalah mikroorganisme yang non aerobik. Oke, nah, there are many microorganism that role as agent of fermentation. Lactobacillus bulgaricus, Rhizopus stolonifer, Neurospora cytophila, Saccharomyces cerevisiae, Streptococcus lactis, and Penicillium regueforti. Yeah. I think all of you have eat yogurt, tempe, oncom, tape ketan, alcoholic beverages, butter, and cheese. Ya, ini adalah this is starter culture of Rhizopus oligosporus, and this is a yogurt from Lactobacillus bulgaricus. So Lactobacillus bulgaricus fermented milk into yogurt. Not fermentation to make lactobacillus, but lactobacillus fermented milk into yogurt. Yeah. And how about food fortification? Food fortification is an effort to add or increase certain nutrients to food that are not naturally contained in the food to improve food quality. Jadi fortifikasi itu upaya untuk menambah gizi di dalam suatu bahan makanan sehingga dia kualitasnya meningkat. Bisa dengan penambahan vitamin D, vitamin B, atau mungkin yang lain. Ya. Jadi istilahnya fortification. Fortification can also be done to restore a nutrient that is lost in the processing of food or beverages. Kayaknya ininya mau habis. Ya, enggak ya? Oh ya. Nah, about fortification, there are some example research. The natural production of vitamin B12 in tempe becomes great, in particular for vegan and vegetarian, since it's usually found from animal-based product. Kidd and Bisping, 1994, showed that Citrobacter friendly and Klebsiella pneumoniae show the best capabilities to produce vitamin B12 in tempeh. However, these bacteria are not generally recognized as safe. Although a study conducted by Ayu, Suwanto, and Barus found that the Klebsiella pneumoniae isolates from Indonesian tempeh were genetically different from pathogenic isolate, some of their strains are still opportunistic pathogens. In a study of Walker, Roy Jacker, Endika, and Smith, so that Propionibacterium fredericii, a grass, vitamin B12 producing bacterium, was combined with Rhizopus oryzae to produce vitamin B12 enriched in lupin tempeh. Propionibacterium fredericii is now utilized to naturally fortify tempeh produced with vitamin B12 in Netherlands and Belgium. It's shown by Istifarah 2020. Istifarah is my daughter. Ya, jadi ceritanya begini. Di Indonesia, tempe yang ada itu sudah mengandung vitamin B12. Kenapa? Karena di dalam raginya itu sudah ada jamur yang ini tadi, yang pneumonia ini, Klebsiella pneumoniae, tetapi strain lain ya. Karena kalau yang strain satunya itu menyebabkan pneumonia, penyakit paru-paru. Nah, kalau strain yang ini itu justru bisa meningkatkan vitamin B12 yang ada pada tempe yang ada di Indonesia. Nah, sementara Klebsiella pneumoniae ini tidak bisa tumbuh di Belanda dan dia termasuk mikroorganisme yang dilarang, ya, yang grass, ya. Jadi tidak safe, tidak boleh dimakan. Nah, akhirnya mereka mencari mikroorganisme lain yang bisa menghasilkan vitamin B12, yaitu adalah Propionibacterium fredericii ini. Jadi untuk menambah vitamin B12 di dalam ragi tempe mereka di Belanda atau di Belgium itu harus ditambah bakteri Propionibacterium fredericii. Seperti itu. Ya, jelas? Ya. Oke. Oke. Aspect of industrial microbiology, there are the dynamic of fermentation, a tool for fermentation, growth kinetic, product download, industrial waste handling, product waste handling, metabolite production, and single cell protein. Jadi kalau kita mau membangun suatu industri mikrobia yang berdasarkan mikrobia, maka harus disusun dulu, direncanakan dulu beberapa hal yang tadi saya sampaikan. Oke. The advantages of microorganism as industrial raw material. Microbes grow quickly. Jadi kalau mau menjadikan suatu mikroorganisme itu menjadi salah satu bahan dalam industri, nah mikrobanya itu harus yang grow quickly. Does not require a large area. Jadi pastikan dia tidak membutuhkan tempat yang cukup luas ya. Dan ini keuntungannya tidak not influenced by climate and easy to control. Tidak terlalu dipengaruhi oleh cuaca dan dia mudah dikontrol. Kemudian juga can grow on various ways that have low economic value to be converted into material with high economic value. Jadi dari limbah pun kemudian dia bisa ditambahkan dan kemudian bisa berubah menjadi suatu produk yang bermanfaat. Seperti itu advantage-nya. Jadi keuntungannya menggunakan mikrobia. Dan yang keenam, di dalam suatu reaksi in a reaction it is necessary to use microbe. Itu apa ya? Memang suatu reaksi itu tidak bisa berlangsung kalau tidak ada mikrobanya. Biasanya seperti itu, ya. Ya termasuk fermentation. Fermentation nggak akan ada kalau tidak ada mikrobanya. Seperti itu. Ya. Nah, ini fermentation media. Jadi medianya, compositionnya carbon source, nitrogen source, mineral growth, and precursor. Ya, ini beberapa cara untuk mendapatkan mikroorganisme ya. Jadi memang suatu produk itu hanya akan terjadi dengan sesuai dengan mikroorganismenya. Ya, kalau kita ingin suatu produk yang good product ya kita harus find a specific microorganism. Ya, seperti itu. Nah, sekarang kita bicara plant growth promoting rhizobacteria. Sebenarnya kalau kita bicara PGPR, it's a large field ya. Jadi banyak sekali yang bisa didukung oleh PGPR. Jadi di sini saya sampaikan plant growth promoting rhizobacteria are the rhizosphere bacteria that can enhance plant growth by a wide variety of mechanism like phosphate solubilization, siderophore production, biological nitrogen fixation, dan lain sebagainya. Ya, nanti materinya bisa dibaca. Nah, yang penting yang perlu diketahui that PGPR is able to increase plant growth, which means increasing plant metabolic processes so that plants are able to produce better or more metabolite. The plants, ini hasil penelitian saya ya. The plants spread with phobio, a kind of biopesticide, a kind of PGPR, were sweeter than those that were not sprayed with phobio. Plants sprayed with phobio can produce higher vitamin C nutrient than plants that are not sprayed with phobio. Jadi tanaman jeruk pamelo yang disemprot dengan phobio, salah satu PGPR, itu dari hasil analisis kandungan gulanya lebih tinggi dan kandungan vitamin C-nya lebih tinggi dibanding yang tidak disemprot. Jadi adanya PGPR ini meningkatkan pertumbuhan tanaman, metabolisme tanaman, sehingga mereka menghasilkan gula dan vitamin C yang lebih banyak daripada yang tidak disemprot. Nah, itu juga one of the role of microorganism. Dan kalau di bidang perlindungan tanaman, penyakit tanaman khususnya, they can increase resistance of the plant. Ya. Nah, this is the graphic histogram. Ya. The orange one is the sprayed with phobio, concentration 10 ml. Ya. Jadi semua kultivar yang disemprot dengan phobio menunjukkan gulanya, kandungan gulanya lebih tinggi. Jadi jeruknya lebih manis. Juga di sini vitamin C-nya lebih tinggi. Nah, yang terakhir, microorganism genetic engineering for increasing food quality. Golden rice. Do you have heard about golden rice? Ya, sudah pernah dengar tentang golden rice? Pernah, oke. Ya, jadi di dalam di dalam tanaman padi itu kandungan vitamin A-nya tidak terlalu tinggi. Kemudian direkayasa sedemikian sehingga tanaman tersebut bisa menghasilkan vitamin A yang lebih tinggi. Meskipun untuk saat ini penelitiannya masih berlangsung. Dari saya itu dulu, silahkan respon dari kalian. Ya, monggo. Terima kasih. Thank you.
Okay, Dr. Wiwit. The next speaker about microbiology will be delivered by Dr. Nur Afiza Badarudin. She is a lecturer from Faculty of Bioresource and Food Industry, University Sultan Zainal Abidin. She graduated in Biotechnology, UPM in 2006. And he achieved master degree in biotechnology from Universiti Malaysia Terengganu in 2011 and PhD in microbiology and molecular biology from Kobe University, Japan in 2015. She is teaching on microbiology, cell and molecular biology, moral and ethics, animal microbiology, and plant pathology. Without further ado, I would like to invite Dr. Nur Afiza Badarudin to present 40 minutes with the title Biological Function of Trichoderma spp for Agriculture Application. Dr. Nur Afiza, time is yours.
Okay, thank you very much, Mr. Moderator. Terima kasih banyak. Assalamualaikum dan selamat petang semua. Masih okey ke semua? Ke ada yang nak mengantuk? Clear tak suara saya?
Dengar.
Dengar Okey, bagi menyegarkan semua orang, saya buat poll dulu. Boleh semua involve sekejap. Okey, link ada dekat chat box dan boleh scan this QR code and siapa yang tak dapat scan boleh pergi ke slido.com, masukkan nombor ni. This code. Please join this poll. Saya just nak tahu apa your first impression, first thinking about micro. Kecil. Dah dapat masuk ke?
Semua mahasiswa sudah bisa masuk? Sudah?
Okay, link ada kat chat box. Boleh check kat chat box.
Di chat box ada untuk mahasiswanya luring bisa langsung di scan barcode di depan.
Apa yang first anda fikir bila dengar je mikrob, apa yang anda fikir? Microbe is a microorganism. Okay, saya cukup untuk ni. Okay, next question. Small living things. Thank you very much for your response. One more question. Okey, sebagai pelajar pertanian, kulat atau jamu apa yang anda tahu? Sorry. Anda tahu. Which one you tiram? More to edible lah. Lagi? Cek kedema. Zainal. Namanya jawab tak kedema ni. Okay. Very good response. Okay.
UPNVJT pun masuk.
Itulah, UPNVJT pun jadi kulat dah.
Aduh.
Okay. So the largest ones is a trichodoma. So many of you know about trichodoma. Okay, thank you very much of your response. This just to interact with you all. Okay, let's go to okay. So ramai jawab trichodoma tadi kan? Okay, today my talk about biological functions of trichodoma species for agriculture application. Before this, docent from Universiti Veteran Jawa Timur talk about functions microbe. It's a more general lah, microorganism in food. Apa function dia dekat food. Tapi untuk saya more to focus fungsi-fungsi biologi of trichodoma, only trichodoma for agriculture application. Basically, this is my research here with my master student Nur Aisyah. I introduce a little bit about my study. As you know, trichodoma is a filamentous fungus. It's a microorganism lah. Mesti tengok melalui microscope, and it produce a lot of secondary metabolites and very one of a beneficial microbes. Okay, so far trichodoma already found as a biocontrol agent, biodecomposer, and plant growth promoting fungi. And in my research, I will prove all these functions. Okay, actually, why we need to study microbe? Why we study microbe? Nowadays, we are terlalu bergantung kepada pesticide, chemical pesticide, synthetic fertilizer, and most of farmers overusage, terlalu menggunakan dengan terlalu banyak, penggunaan yang banyak tanpa mengikut dos yang betul, tanpa mengikut yang sepatutnya lah. Penggunaan yang berlebihan akan menyebabkan berlakunya pollution terhadap soil itu sendiri. Jika soil adalah foundations of agriculture, kalau tak ada soil memang susah. Memang ada sekarang ni dah ada hidroponik, tapi still lagi soil adalah sumber utama untuk agriculture. Memang dia punya asas. Bila penggunaan yang terlalu tinggi synthetic fertilizer dan chemical pesticide menyebabkan acidity tanah tu menjadi tinggi dan menyebabkan banyak kerosakan berlaku pada tanah tu. Kadang-kadang terlalu banyak fertilizer digunakan boleh menyebabkan ketidaksuburan tanah, dan bila terlalu banyak pesticide digunakan menyebabkan banyak mikrob yang berfaedah akan mati. Sebab chemical pesticide agak toksik kepada mikrob-mikrob dalam tanah, mikrob-mikrob natural mikrob. Dan okey, disebabkan saya akan sentuh juga pasal decomposer, memang masalah sekarang ni ialah food waste yang berlebihan, dan terlalu banyak pembaziran, terutama di pasar raya jika sayur-sayuran tak habis, di hotel makan tak habis, even di rumah sendiri. Katanya kita boleh amalkan, kita kompos sendiri food waste kita jika adanya bantuan-bantuan mikro ni. Okay, today I will talk about 3 functions of trichoderma as a biological control agents, biodecomposer and plant growth promoter. Okay, saya akan terangkan sedikit pasal trichoderma ni. Trichoderma ni non-pathogenic fungi. So far saya dah test dengan beberapa host, dia okey. Tidak memberikan kesan yang buruk pada host yang kita test. Kebanyakan disease ialah dia more spesifik. Dia spesifik pada pokok-pokok tertentu. Okey, dan dia mengeluarkan banyak secondary metabolites. Contohnya peptabols ni dia akan membantu inhibit cell wall, inhibit beta glucan synthase enzyme, menyebabkan dia boleh memberhentikan, mengganggu pertumbuhan patogenik fungi yang lain. Nanti kita, okey, contoh kat sini. Plant disease management of trichoderma species yang telah dikaji ada 3 melalui jika ada fungus menyerang pokok, dia akan serang balik, atau dia melindungi pokok itu dengan metabolite yang telah diproduce, atau jika ada nematode yang tidak bagus untuk pokok tu, dia akan menyerang nematode tersebut. Ini adalah cara yang telah dikaji bagaimana trichoderma tu menyerang pathogenic fungi secara coiling atau formation of appressorium, di mana dia akan membentuk appressorium untuk menyebabkan sel wall pathogenic fungi itu akan rosak. Untuk detail explanation, you can refer this our paper. This is a review paper, biological function of trichoderma species for agriculture applications. Okay, as a biodecomposer agent, a lot of paper, not a lot, still not a lot. Kalau di Malaysia ni pernah study kepada EFB atau POM. EFB is an empty fruit bunch. Untuk yang tandan sawit yang telah dikeluarkan buahnya, untuk kompos, untuk kita boleh jadikan, bila kita kompos dengan micro, dia akan reduce the composting time. Dia kalau kita tak ada inoculate fungi, take about 4 to 6 months to compose, but with trichoderma only 1.5 months to compose. Same with palm oil meal effluent. Dan jika kompos ni diinoculate dengan fungus atau bakteria, dia akan menambah lagi value kepada untuk dijadikan sebagai biofertilizer yang boleh digunakan balik kepada pokok-pokok sawit. Okey, untuk sebagai plant growth promoting fungi, PKKM ini memang dikaji trichoderma untuk spesies yang dalam study saya ni, so far memang banyak trichoderma. Contoh yang dalam saya ambil dari paper ni ialah trichoderma virens. Dia memang ada satu auxin, ada hormon yang boleh membantu menyebabkan ada cell elongation, cell expansion atau cell growth lah. Menyebabkan cell root atau cell shoot tu akan lagi cepat compare to tidak ditambah dengan mikrob lah. So sebenarnya kita boleh mengurangkan kebergantungan kita kepada synthetic fertilizer dan menyebabkan, sebab so far lah bagi saya trichoderma je lah. Mikroorganisma ni cost dia lagi murah daripada synthetic fertilizer. Okey, untuk method bagi yang boleh, ada yang ambil bioteknologi pertanian, ada yang ambil crop science, ada yang tapi yang saya tahu untuk mahasiswa yang hari ini ialah kebanyakannya memang base dia pertanian lah kan. Okey, ada yang kalau ingin mengkaji certain microbe, saya akan terangkan very brief about the method of how to test the microbe. Ada fungsi tak? Ada fungsi as biocontrol agent or as plant growth promoter atau biodecomposer lah. Okay, first usually I will ID first my fungus lah and screen for antagonistic activity. Okay, untuk screen for antagonistic activity actually sebenarnya banyak assay yang boleh dilakukan, tapi the easiest is dua culture assay lah. Kita just put the different fungus out. One is our tested fungi, and one fungus, and one is pathogenic fungus. So we check the inhibition zone. And for that biodecomposer, selalunya kita akan screen dulu apa enzim yang ada. Enzim apa? Kita screen dulu enzim apa yang nak, enzim apa yang ada, dan kita start boleh analyze dia. Okay, untuk fungi ni enzim ni ada, untuk fungi ni enzim ni ada. Okay, dan kita try untuk compose beberapa material lah. Kalau dalam my research compose food waste, ada yang my student compose banana trunk, ada banana peel. I will show you the results. For today, I just want to share my research. Okay, these are enzymatic activities of trichoderma isolates. For this study, I screen enzymatic activities for 5 substrate: lignin, cellulose, starch, xylene, and pectin. Because these 5 substrate is substrate for cell wall. So kalau trichoderma dapat mendegradekan these 5, usually they can degrade cell wall of food. Sebab kebanyakan raw material daripada yang food ialah daripada tumbuhan juga kan. Okay, just for check the enzymatic activities, just grow the trichoderma on the plate that contain the substrate. Okay, and then I try to compose with a different trichoderma and check for plant growth promoter. First, we check is it contain IAA or not. IAA is one of auxin hormone. And check treat the seed with trichoderma, and I try to treat the soil with trichoderma. I will show you the result after that. Okay, for IAA, we use a Sapolsky reagent and check the absorbance. And for we treat the seed for this study. Actually, my student, different students, I mean, test a different crop. Ada yang guna pak choy, ada yang guna okra, ada yang guna melon. So I will show you the result after this. Okay. Actually, for my research, I use 3 different trichoderma species. Hamatum, Trichoderma hamatum, Trichoderma hasianum, dan all these trichoderma already molecular ID and we confirm all the identification. Okay, for identification, you must do molecular and morphological. For morphological, you can check the structure, the concentric ring, and check the microscopic feature, conidia, spores, and hyphae, and the have a septa or not, the mycelia. And you can check with the book, ID book. Okay, then from our research lah, trichoderma are very good on have a very high antagonistic activity against a lot of pathogenic fungi and bacteria. These are some of these are, you can see a Coleotrichum truncatum, Coleotrichum catsky, Coronaspora cassicola. The Coleotrichum cause the anthracnose disease, right? And Coronaspora cassicola cause the CLSD. I think for the pertanian student, agriculture student, I think this fungi is familiar, right? This plant pathogen. And we can see the trichoderma can inhibit the growth of all these pathogenic fungi. All the trichoderma are the selected trichoderma, these three species. Same with bacteria. Okay. Also, my student also test with insect because di sebahagian utara Malaysia, we got a problem bedworm. You know bedworm, Metisa plana. Bedworm that infect the oil palm. These are oil palm bedworm lah. This picture show the bedworm. We test a trichoderma. Just spray the trichoderma on it, and the mortality rates in 7 days almost high. For trichoderma atrovide is almost achieve 100% in 7 days. Quite good. And toxic to bedworm. Okay, for biodecomposer, after test, like these three selected, the three species of trichoderma show very good, very good enzymatic activities. You can see here the degradations of cellulose here. You can see the cellulose. For lignin is a browning reaction, like you can see here. For cellulose, you can see from the red color become the purple color. All the substrate is degraded by this fungi. Okay. Actually, we also test to food waste. That I show you the certain our research output. Okay, this banana peel. You can see turn into liquid in just 2 weeks by using a trichoderma. Turn this banana peel, this a banana chunk. It's reduce the composition time. Then we check the nutrient, very high content of phosphorus, calcium, sodium, and magnesium. So these all compost can use as biofertilizer. Very good for plant growth. Okay, for plant growth promoter, we check the IAA. The trichoderma is quite high in IAA. So it's a proof that trichoderma can help in plant growth. Okay, these are my student results. Use a pak choy as a tested plant. you can see this a seed treatment, not the soil treatment. We treat the seed before we germinate. We just treat the seed with the fungi, and you can see the root length is different from the control. The root length is significant longer than a control. And for consortium, and surprisingly, consortium, we combine all the 3 fungus. The root mass is not too long, but the root mass is lebih banyak lah, lebih banyak. You can see the root dia bukan panjang, dia lebih banyak. You can see in the picture, right? Okay, untuk kami cuba juga untuk treat dengan soil. Kami cuba treat dengan soil, dan Hazianum show a very significant difference from others untuk plant head, root mass, root length, leaf area, chlorophyll content, and root length. So you can see here, even the number of leaves is different from others. Okay. Dengar kan saya? Dengar tak? Kenapa sunyi sangat ni?
Dengar.
Dengar kan? Okay. Kami juga check contents jika tambah dengan trichoderma, ada beza tak dia punya contents nutrient contents dia. Bila kita check memang ada beza. Nitrat dia lagi tinggi, potasium lagi tinggi, kalsium, ferrum and manganese. Yang tu memang sangat membantu. Mana trichoderma sangat membantu untuk menguraikan nutrien-nutrien dalam tanah tu, untuk senang lagi plant tu untuk uptake. Okay, so the conclusion is trichoderma ni memang fungsi lah. Memang, so kita boleh lagi kaji mikroorganisma yang lain untuk membantu dalam pertanian ni. Bukannya so kebergantungan kita kepada sebenarnya produk berasaskan mikrob ni still lagi kurang dalam pasaran, dan petani sangat bagi saya tidak memandang berat lah benda ni bagi saya. Sebab even trichoderma ni, even satu fungus pun dah boleh bagi function yang as a biological control, as a biodecomposer and as plant growth promoter, tapi still lagi sangat kurang diberi perhatian. Kalau saya still Malaysia lagi masih kurang lah memberi perhatian kepada produk-produk yang berasaskan biologi ni lah. Disebabkan penggunaan yang berlebihan, penggunaan pesticide, terutama pesticide lah, sangat memberi impact kepada pest ni lah, pest and disease ni. Bila terlalu bergantung pada pesticide, kebanyakan pest and bacteria and fungus, pathogenic fungus ni akan menyebabkan dia lagi resistant. Jadi bila saya tanya farmers, diorang terpaksa guna lagi banyak, dos yang lagi tinggi, kadang-kadang guna yang lagi kuat. Sebab apa yang berlaku? Sebab bakteria-bakteria atau fungus ni telah resistant. Kepada pesticide tersebut. So sangat tak bagus untuk akan datang lah. Dan menyebabkan benda tu kan impact juga kepada manusia lah. Chemical semua tu sebenarnya bila kita makan pun still ada residu di dalam plant-plant yang kita makan tu. Kan ada juga yang farmers nak jalan mudah, dia guna racun-racun yang telah di ban, yang tidak boleh. Maknanya yang memang meninggalkan residu yang banyak dalam plant tu. Sebab tu bila sebenarnya itulah, tapi benda tu more to educate the farmers lah. So pelajar-pelajar pertanian sekarang sekalian ni boleh try to educate the farmers sekarang ni lah. Lagi kita dah more to modern agriculture kan. Kita mesti dah start bergantung pada bio-based product lah untuk mengurangkan chemical pollution. Dia bukan kepada tanah sahaja tapi juga kepada manusia lah. Impact juga kepada health kita, kesihatan kita. Okay, this is our product from this research lah. The name of Tri-Compact. Tri tu is from trichoderma lah. We prepare as a tablet for this product, and we use all the material where they tested trichoderma from this product to test as a treatment.
Multifunctional byproduct towards sustainable agriculture. As stated in the news, we can recognize some of the agriculture problems, which are excessive use of chemical products, pathogenic fungi, and food waste. Chemical products such as chemical fertilizer and chemical pesticide lead to fewer or poor quality crops in the long term. This is because of the intricacies of soil health. Like humans, the soil needs a delicate balance of nutrients to remain healthy. Simply adding chemical products to the soil without regard for keeping the balance can lead to unintended consequences or hidden dangers. In addition, chemical products can make the topsoil acidic because it works towards the pH of the soil. The idea for maximum plant growth and crop yield is between 5.5 and 8. If the soil is too acidic, it will yield less crop. Plant pathogenic fungi. If the crop are infected by plant pathogenic fungi such as Fusarium species, Collectotrichum species, Rhizoctonia species, it will decrease the yield of crops. Food waste. According to Lim et al. In 2016, the management of food waste in Malaysia usually not effective and costly compared to other Asian countries. The food waste throughout Malaysia will be gathered in their respective landfills and required months and years to decompose naturally. The concern is landfill produce toxic leachate into the soil. Hence, it will disturb the balanced nutrients in the soil. To sum up, soil is important to be taken care of and is the foundation of food system. But don't worry, this is where the Tri-Compact save the soil and also do some magic. So, what magic can this Tri-Compact do? As you can see, this magical Tri-Compact can act as a biodecomposer, as a biological control agent, and as a plant booster. Plant cell wall is hard to degrade. As you can see in this illustration, the cell wall consists of cellulose, hemicellulose, xylene, lignin, starch, and pectin. However, Tri-Compact can degrade lignin, cellulose, starch, xylene, and pectin. Hence, Trichoderma hamatum, Trichoderma hanseanum, and Trichoderma atroviridae in Tri-Compact are enzyme producer and as a plant biodecomposer. As a biological control agent, tricompact can inhibit more than 50% towards plant pathogenic fungi. As you can see in this figure, this is the data collected after the treatment. Hence, tricompact can prevent disease as stated in the slide. Finally, as a plant booster, you can see the effect of plant growth referring this figure. Without tricompact, the plant will exhibit small diameter of leaf with short root and less root formation. Whereas with tricompact, the plant will exhibit positively large diameter of leaf with larger bulky root. Let's imagine the plant root as your hand. If you have short and small hand, you are limited to collect small amount of nutrients close to you. Whereas if you have long and bigger hand, you can collect vast amount of nutrients. To summarize, the product can move forward toward sustainable food system. Before ending this video, I would like to recap the magic of tricompact. Over-reliant use of chemical pesticide in agriculture will affect environment, pollution, and beneficial insect or plants. By using tricompact, it will save the environment and pollution. Compost that treated with trichoderma species or tricompact contribute to highly nutritious biofertilizer. This product guaranteed the potential biological approach for sustainable disease management in Malaysia. Thank you for your attention. Bye.
Okay, that's all from me. Thank you very much.
Thank you, Doctor. All right, I'm sure everyone have a question to Dr. Wiwit and Dr. Afiza. We will have 20 minutes for discussion. Please, our participant. Do you have any question to Dr. Wiwit and Dr. Afiza about microbiology? I think in column chat, hi, I'm Hanani from Unisa. I want to ask to Dr. Afiza, is there any requirements to culture Trichoderma spp? Question again? Any question again? I think Dr. Wiwit can answer. Yeah.
If the PGPR cannot increase the resistance, it means that maybe the PGPR is not suitable for that plant, or maybe the second question, they are not can grow better or cannot grow because maybe the climate or maybe the medium is not suitable for them. Jadi PGPR itu kan juga membutuhkan medium dan cuaca atau climate yang sesuai. Kalau kemudian dia ditumbuhkan di dimasukkan ke dalam suatu lingkungan untuk membuat tanaman tadi tumbuh dengan baik, kalau lingkungan di situ tidak sesuai, kemudian mungkin tanahnya juga tidak sesuai, bisa jadi they cannot grow better. Ya. Sehingga pertumbuhannya tidak bagus, otomatis dia tidak bisa memberikan benefit to the plant. Jadi ini nyambung ya pertanyaannya yang pertama dengan yang kedua. Kalau ingin menambah benefitnya, berarti ya sejak awal mediumnya untuk pertumbuhannya itu dibuat sedemikian sehingga dia bisa cocok mediumnya, sehingga dia bisa tumbuh dengan baik dan lingkungannya juga mendukung. Karena kalau kita ingin menggunakan suatu mikroorganisme kan harus ditumbuhkan dulu. Dan itu butuh waktu, butuh medium yang cocok, dan butuh lingkungan yang cocok. Itu memang kesulitan ketika kita menggunakan PGPR atau agen hayati. Seperti itu. Ya. Ya.
Is that clear? Sudah jelas? Okay, Dr. Afiza, maybe you can answer.
Okay, untuk soalan daripada Hanani, thank you Hanani. Okay, untuk requirement untuk culture trichodoma, untuk trichodoma dia tak adalah susah, dia tidak memerlukan suhu yang sangat. Dia range dia lebih kepada suhu bilik pun, macam suhu kita tropikan tak ada masalah pun. Kita boleh grow tanpa guna incubator. Selalu student saya hanya grow kat room temp je. So untuk culture dia, so far kita dah test because kita produce product, kita test untuk reduce the cost dan kita test untuk grow dia balik, just guna starch asal ada carbon source, still okay untuk grow dia. As long as ada spore dia boleh regrow balik bila kita mix dengan tanah atau mix dengan media. Dia memang boleh regrow balik. So untuk yang kita guna sebagai pemegang dia, untuk yang produk kita tu, kita just guna starch, glucose, kaolin. Starch, just ada carbon source lah. Itu yang penting. Sebab karbon kan dia punya nutrient intake yang sangat penting lah untuk microbe. Okay, that's all.
Bu Wiwit akan menambahkan.
Kemarin saya penelitian di 2 lokasi yang berbeda. Yang satu itu tinggi tempatnya, yang satu rendah. Nah, ternyata Fobio PGPR yang saya gunakan itu dia bisa tumbuh dengan baik di lingkungan yang di bawah, yang rendah, ya. Dan itu hasilnya yaitu bahwa setiap minggu itu selalu tumbuh tunas-tunas baru recovery dari setelah dia diserang oleh fusarium. Jadi tadinya diserang fusarium, disemprot Fobio, kemudian dia muncul tunas-tunas baru setiap minggunya itu recovery. Tetapi yang ada di payangan itu beda, tidak sama dengan yang ada di celuk yang ada di dataran rendah. Nah, barangkali ini juga merupakan suatu faktor penentu bahwa lingkungan yang di celuk ini cocok untuk PGPR yang ada di Fobio. Kebetulan Fobio itu PGPR-nya itu dari rhizosfer, tanaman tebu, sugarcane, kemudian coconut, kemudian mangrove, dan siwalan. What is siwalan in English? Ya, siwalan. Nah, itu kan daerah dataran rendah ya, sehingga mungkin itu juga mempengaruhi. Seperti itu. Terima kasih, Mas Ari.
To Dr. Afiza from Dr. Arika Purnawati. In your presentation, I saw the presence of Serratia bacteria. Is this bacteria a plant pathogen and also a human pathogen? Is it right?
Yes. Serratia also human pathogens. But from my study, I isolate bacteria. I isolate this Serratia from lettuce, infected lettuce, and checked by Koch postulate, and Serratia caused this the disease. And I think I already published about the Strepsia cause disease for the lettuce. I already forgot. I will check later. But it's also human pathogen. Yes.
Okay, is there a question again? Class? No? Oh, okay, one question again. Please.
Baik, perkenalkan saya Sinta. Saya ingin mengajukan pertanyaan kepada Bu Wiwit. Saya ada 2 pertanyaan, Bu. Jadi mengenai sertifikasi pangan tadi. Menurut Ibu, apakah sertifikasi pangan yang berkepanjangan tidak akan berdampak pada keanekaragaman plasma nutfah kita? Jadi misalnya beras yang saya cari itu misalnya beras yang diberikan vitamin tambahan gitu ya Bu. Nah kalau beras itu memiliki vitamin yang komplit kan akhirnya masyarakat itu tidak perlu membeli seperti buah kayak gitu. Nah lalu itu apakah tidak berbahaya bagi keanekaragaman plasma nutfah kita? Kemudian yang kedua, sertifikasi pangan tidak memiliki potensi yang berdampak negatif terhadap kesehatan manusia, seperti yang terjadi pada transgenik yang memicu kanker pada manusia. Begitu. Terima kasih, Bu.
Baik, terima kasih. Ini arahnya kepada fortifikasi yang genetic engineering ya. Ya, jadi yang menggunakan GMO. Memang sampai saat ini masih banyak polemik tentang GMO ya, karena memang apapun ada dampaknya. Salah satu yang pernah saya alami saja itu adalah alergi. Jadi ketika saya makan kedelai yang dari GMO itu langsung gatal semua di badan, dan itu saya nggak tahu apakah orang lain juga mengalami hal yang sama. Jadi memang GMO sampai sekarang itu masih mengalami polemik ya, antara diterima dan tidak. Dan itu kalau di sana di Amerika itu jelas sekali perbedaannya, siapa yang mengikuti GMO dan siapa yang tidak. Dan biasanya yang orang-orang yang condong ke sustainable agriculture, sustainable development, organik, itu dia nggak mau menerima GMO. Ya, memang di kita pun saya kira juga seperti itu ya, masih banyak juga polemik tentang GMO. Tapi saya di sini tidak ingin berbicara tentang polemik tersebut. Ya, kita bicara tentang ilmiahnya. Monggo nanti kalau masalah polemik kita serahkan sama pemerintah atau mungkin yang berwenang. Saya kira itu ya, Mbak ya. Ya.
Okay, I will close. Question. Okay. Perkenalkan, nama saya Muhammad Effendi, asal dari UPN Veteran Jawa Timur. Ingin bertanya kepada Gubernur, dengan cara apa atau mekanisme apa Hipotherma dapat menjadi GPR? Terima kasih. Afrika benar tak?
Tak jelas.
Tak jelas.
Saya dengar yang depan je, belakang tak dengar.
Alright. Cara apa atau mekanisme apa Hipotherma dapat menjadi GPR? PGPR.
PGPR.
So macam mana trichodemo boleh jadi PGPR?
PGPR.
PGPR.
GPF lah, dia plant growth promoter fungi. Dengan cara apa, mekanisme apa kan? So far yang kita kaji ialah dia boleh hasilkan hormon lah, hormon auxin. IAA dia agak tinggi. Yang tu mekanisme yang telah dikaji lah so far. Dia memang sangat membantu dan dia boleh mengurai bila kita check dengan ICP OES, kita check nutrien tanah tu memang ada pertambahan. So memang ada beza dengan kontrol di mana tidak diletakkan trichoderma. Memang ada pertambahan setiap nutrien yang saya sebut lagi, kalsium, manganis, ferrum, apa lagi, fosforus sangat tinggi dalam tanah yang mengandungi trichoderma. So dia juga membantu pokok tu ambil nutrien-nutrien tersebut, selain daripada dia membantu memanjangkan sebab dia ada auxin tadi, dia ada IAA tadi yang tinggi, dia membantu untuk cell elongation, untuk root dan shoot, tunas dia dan akar dia. Kan tadi saya tunjuk contoh akar untuk yang kita treat dengan trichoderma memang lagi panjang. Tu memang very significant lah. Dan bulky, dan dia root mass dia lagi banyak. Yang tu dia boleh berfungsi sebagai PGPF.
Okay, is that clear? Sudah? Okay. It's okay. Okay, so I will close off this session microbiology. To our speaker, to meet you here in short time, surely not enough. I hope we can arrange for the collaboration for research or exchange student for our student to study more about agriculture between Indonesia and Malaysia. We do hope Dr. Nalini, Dr. Salma, and Dr. Afiza always be healthy. And we will welcome you if you visit our university in UPN Surabaya. Before it ends, we will give a certificate to 6 speakers from University Zainal Abidin and UPN from appreciation from us. Please, committee, to share certificate. This first certificate to Associate Professor Dr. Nalini Arumugam, not Arumugram, not using R, please.
Spelling mistake.
This typo.
Without R.
Okay, next. Okay, it's okay. Next. Certificate of appreciation presented to Dr. Salma Mohammed. As a guest speaker. Next. The certificate appreciation to Dr. No Afiza Badaluddin as guest speaker from University Sultan Zainal Abidin. Okay.
Thank you.
Welcome, Doctor. And this is certificate of appreciation presented to Dr. Randa Sutini MPD as guest speaker from UPN Veteran Jawa Timur.
Alhamdulillah, thank you very much.
And this is certificate of appreciation presented to Ramadhani Mahendra Kusuma SPMP MSc as guest speaker from UPN Veteran Jawa Timur. And the last certificate appreciation presented to Dr. Insinyur Sri Wiyatiningsi MP as guest speaker from UPN Veteran Jawa Timur.
Thank you.
Okay, we already reached the end for global class. Thank you so much to all our amazing speakers for addressing interesting topics. And thank you to the audience for your participation. I hope you have learned and enjoyed the presentation and the World Global Class program. Let me also take this opportunity to thank and congratulate the organizer. After the first event done, now we are going to the last agenda. There is a closing. But before we close this event, let's make beautiful memories by taking pictures together. Okay, all of you guys,
Can you turn on your camera, please? Maybe committee can screenshot first. Yeah. Okay. Yeah. Okay. The first page. 1, 2, 3 is. Okay. Next. 1, 2, 3 is. Okay. Page 3. Nggak ada yang nyalain kamera ini. Oke, 1, 2, 3. Halaman 4. 1, 2, 3. And last. 1, 2, 3. Today event ends. I am Haidar Fari Aditya as the moderator, a lecturer from UPN Veteran Surabaya. I'm sorry if there was any mistake when guiding the Global Class event. Very thank you to all participants in this class. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you all.
Thank you very much. Thank you very much. Take care. Bye.
Yes, Doctor.
Thank you. Thanks a lot. Take care.
Jumpa lagi.
Jumpa lagi.
Bye. Jumpa lagi.
yang